Hello, everyone. We're about to start with our very next session, but as everyone is getting to their seats, I just have one quick announcement that if Jean-Giacomo Navarra is here, your Holiday Inn room key is at reception right outside, so you can pick it up whenever at the break. And I'm going to turn it over to Victoria to introduce our next session.
Thank you. Welcome, everyone. I am so excited to introduce our next session named Coupled Biogeochemical Cycles, Interconnected Controls on Ocean Fertility. And we have a series of great speakers and two sets of lightning talks. And we're not going to have a long introduction. I'll let the great illustration speak for itself. And then I'm going to hand it over to Alessandro Tagliabue to give us an introduction talk.
Okay, while we get set up and before the timer starts, I'll start talking already. So I want to thank the organizers for putting together this really exciting session and for inviting me to speak. I found it strange to realize yesterday that it's like my first OCB summer workshop, but then that seemed a bit weird since I feel like I know Heather, May, Mary very well, and I guess it just shows that I've been interacting with a lot of the other activities that OCB has been involved in over the years. So I think that's an interesting kind of observation, and I think it's a great arena, a great crowd, and I'm sure it won't be my last OCB. I'm going to start off this session with quite a broad title, quite an ambitious title, and I'm going to borrow some of the words of the title, and I'm going to home it in a little bit, and I want to talk about biogeochemical cycles, ocean fertility, which I'm going to take as productivity of the ocean, and how it may operate in a changing climate. There's a few colleagues there that I'd like to acknowledge who've made important contributions to the work I'm going to show. So, okay, just the old-fashioned way. So the first thing I want to do is start us off with the kind of grand challenge of what we're worried about trying to do, and that's to provide useful forecasts in terms of a changing climate of how our ocean is going to change in the future. Then I want to home in on a case study about how shifting patterns of nutrient limitation may be a really important driver of uncertainty in these predictions, forecasts, maybe is the best word. And then I want to close by giving you an example of where we've tried to develop what's called a kind of emergent constraint on model behavior, which may allow us the potential to sort amongst Earth system models to hopefully end up a little bit more confident about what's going on. Then if there's time, I've thought of some interesting questions that I couldn't fit into the talk that can act as a kind of caveat to things I should have mentioned and may also stimulate some discussion. So this topic, coupling the cycling of elements in seawater with biological activity, it's not a new topic, and we're in this building named after a very famous oceanographer, and this was right in the kind of the big man sort of realm. So I start really with this I think fantastic paper written by Redfield back in 1958. If you haven't read it, I really recommend reading it, and it is actually still very relevant today to some of the key questions that we're trying to answer. And I'll draw out even just one term, this concept of the biochemical circulation, right? The movement of material and bioessential elements through the biology as a kind of a circulation scheme in the ocean. And so that's your homework. Go and read that. And this is a really nice figure that was put together in the paper that tries to illustrate this concept of biology on this side, essential elements on this side, and this very close tango that they're doing together. The one relies on, biology obviously rely on essential elements, but then how they take up those essential elements, how those essential elements get recycled, imprints also on their availability in seawater. So I think this is still very influential, very useful framework. It's so useful I've sort of simplified it with my best PowerPoint kind of clip art skills into a green blob, some arrows, and I've tried to highlight the essential elements here. And now, with apologies to the man upstairs, I'll kind of simplify this even more into a framework where we have a sort of biological reservoir, we have a set of essential elements, they get taken up through a process called productivity, and they get ultimately recycled back into water. And I want to think about this with a little bit more depth. I want to add one more arrow, and that's how these elements get into the environment where the biology are growing, okay? And so originally I had mixing here, but then I didn't want to upset Douglas, so I made it more general and made it inputs, which could be aerosol deposition as well. And what I want to think about is understanding how ocean fertility is going to respond to changes in that environment. You know, we have a very simple view about how changes in this cascade into changes in this on the other side, and that's the challenge I think we need to grapple with as a community. If we take a step back and we do a bit of physics to keep our physical colleagues happy, this is what we produce in terms of information for policymakers, and this is showing you how the sea surface temperature on the y-axis will change in the future as a function of various different climate change scenarios. And they're all the colored lines. And this presents a very, very clear message to policymakers because we see a clear distinction amongst the colored lines, which relate to different emissions forecasts. And thus, you know, the more carbon we emit into the atmosphere, the greater change of SST we see. Okay, so if a policymaker was to walk in, they'd say, brilliant, this is really useful for me. I can use this to motivate to get onto different emissions pathways because of the clear benefit in avoiding levels of warming. If we produce the same information for productivity, okay, suddenly you get a very, very different picture, right? We can't differentiate one line from another. And what was here, the model spread, this little gap here, suddenly, you know, encompasses the entire figure. And now the policymaker is a bit kind of like, you guys don't know anything, right? Because you can't even tell me the sign of the change in productivity. All these different climate models effectively give a very similar answer, which is that productivity is going to change very little, plus or minus an enormous number. Okay, and what that tells us is some models think it's going to go up, some models think it's going to go down. And this is, we can laugh, but this is a serious challenge we have in communication because, you know, this is a challenge, and we have to be able to deal with this because folks need these forecasts, right? If we want to understand the response of marine ecosystems, if we want to give useful information to those who are trying to design marine protected areas, if we want to provide information on fisheries scientists, for fisheries scientists, they need this information. And somehow we've got to do better at this. If we look at a regional scale, so now what I've done is I've calculated that same quantity, but for these different boxes in the ocean. Global is now down here. Now you see what looked massive before is tiny, right? And there's even more uncertainty across the models in different regions, okay? Especially in the tropics. Okay, so what I've done is I've highlighted the Indian Ocean, Equatorial Pacific, and the northern southern Pacific gyres. And around 2/3 of that global uncertainty is explained by the uncertainty in these regions. And it's no coincidence that these are the regions where society relies on protein from the sea, right? So this is like really important for us to fix this. I'm going to talk in this presentation about the Equatorial Pacific, where we have a really large uncertainty across models. So we want to understand this regional change a little bit more. So before we do that, we want to think about what goes into these models, right? I'm not a mathematician, hence I haven't used the fancy mathematics function on PowerPoint, and I've just written it out. I ultimately want to understand how this reservoir is changing in time, right? So that's a differential equation. Rate of change in this as a function of time is, of course, a function of sources and sinks, right? So if we think of productivity as the term that's going to grow this reservoir, and then there's a load of loss processes that, with apologies to those who work on it, I'm not going to focus on too much. So this productivity is actually a very simple calculation. In the models, we compute a growth rate, okay, and we multiply that growth rate by the carbon biomass of the phytoplankton group for which we've calculated the growth rate for, and that takes us from a sort of per day or per second rate into a moles of carbon per volume per second, and that's our rate, okay? That's our productivity term, okay? Note, we don't really model photosynthesis in these kinds of models. So then actually the key becomes this growth rate. And so this growth rate, you know, we still model it in a very simple way that goes back to Riley, Steele, Fasham, the kind of pioneers of like ecosystem modeling. You know, we calculate a maximum growth rate, which we say that, you know, if everything were available in perfect abundance, organisms would grow at that rate, but they don't because they get limited by nutrients, okay? We know that there's many nutrients in seawater, but we neglect all of them bar the most limiting nutrient, okay? And we assume that is the resource that is driving the growth rate. That nutrient, that concern about the most limiting nutrients means that we need to worry a lot about nutrient acquisition. So how are biology taking up nutrients from seawater? What's driving this upward arrow? And that's what I want to focus on in this presentation. So we'll highlight it in yellow. But what I want to do is I want to think about NPP uncertainty in projections of ocean fertility and how they're linked to uncertainties in acquisition of nutrients. And I like iron, so I'll focus on iron. And what I want to do is I want to take one single model, and instead of taking loads of different models produced by different groups, I want to take one model, and I want to force that model by exactly the same climate scenario, a high emission scenario, and I want to vary components of that model and see how that affects the trends in productivity, okay? We're going to focus on the Eastern Equatorial Pacific, because as I showed you, it's a hotspot of uncertainty in the global change. And all we're going to do is we're going to mess around with this simple trait around how good biology is at acquiring iron from seawater. And in the model, that's controlled by two different variables, Vmax, maximum rate of internalization of iron, and the maximum quota. So to what extent can these organisms accumulate iron above that required to store? So we can play around with the assumptions on these two terms, and we can look at what happens to the projected change in productivity, okay? So what we find is actually there's a massive impact, right, when we alter these terms. This is the Eastern Equatorial Pacific. This is the change in net primary production into the future. And the black line is the control model, or our control model, and it predicts a large decline in productivity. And then we've got these different families of simulations. Don't worry too much about the numbers here. They just reduce the size of these terms. So they basically reduce the competitive ability of phytoplankton in our model to acquire nutrients. And you can see actually, you know, if we assume a slight reduction in either the uptake rate or the cellular quota, we can more or less halve the change in productivity. If we reduce it still further, we can stabilize productivity, or maybe it might even go up a little bit in the future. So just by modulating a simple trait in the model that's like very poorly observed, you know, it's very difficult to pick the number, we can generate a really significant amount of variability in the net primary production, which spans what we saw originally, right? This like large uncertainty. So we need to know more about why this is happening, and maybe this has got something to do with what's going on in the models. But unlike nitrogen, iron is the more complicated element. You know, it's controlled by physical supply, biological uptake, but there are other sources of iron to seawater. And biological uptake is not the only sink for iron in seawater. And so what we're showing here is basically the balance between these biological uptake of iron and what's called scavenging, so the abiotic removal of iron due to precipitation, aggregation of colloids, or adsorption onto particles. So you can see in these experiments, what we've done, we've lowered the biological uptake as we wanted to, right? And we've lowered it by different degrees. But what we see is that there's this compensation from the abiotic removal terms. So as we reduce the competitive ability for phytoplankton to take up iron, more gets lost through the abiotic routes. That's important because it affects, you know, in our model and in reality, when biology take up iron, it gets transferred through the food web, it gets recycled by zooplankton, by bacteria in this process known as the Ferris wheel. But as we modulate these traits, we're kind of modulating the ability of iron to be retained in the upper ocean by biological processes or exported. And that affects the patterns of nutrient limitation. So if we take the two extremes, right, a high iron uptake and a low iron uptake, this model simulation showed it was the control, it showed a big decline in productivity in the future. The model simulation on the right showed very little change in productivity. And what we can see is when we're under high iron uptake, as we move into the future, the iron-limited area of the Equatorial Pacific declines quite a lot, and it gets replaced by nitrogen limitation. Conversely, when we have the low iron uptake, this iron limitation is very resilient. throughout the whole simulation. So we don't see this abrupt shift. These shifts are important because these models, the growth rate is driven by the most limiting nutrient, right? So if you keep iron as limiting throughout the whole duration of the simulation, then it is solely responsible for driving the changes in growth rate. If you allow these nutrient limitations to shift, then suddenly, at some point during your experiment, nitrogen becomes really, really important. So if the upwelling declines, it then becomes the driver of NPP. These shifts between nutrient limitation actually are predictive of the change in productivity at the scale of the entire Pacific, okay? These are all our experiments. So you can forget about that. You can just follow, they all follow this same line. The more we change the iron-limited fractional area, the more we drive down productivity, okay? If for some reason we're able to retain strong iron limitation in the future, we stay up here, right? And we have a very small change in productivity. And maybe productivity might even increase slightly in this. If we change the iron-limited area a lot and replace it with nitrogen limitation, suddenly we get the drop in productivity. But this suggests that small differences amongst models about how they apply these physiological traits can kind of cascade into these large impacts on the future predictability of productivity in the region. This isn't just limited to the equatorial region. These things we think might also be happening in the Southern Ocean, where there's potential shifts between iron and manganese limitation. This is just an example from a recent paper we published with an iron and manganese-enabled kind of climate model, which showed the growing manganese limitation under a high emission scenario. These things also happen potentially during the last glacial maximum, where we add iron to the system and we generate manganese limitation. Ultimately, just like before, the extent of these changes, i.e. potentially here the difference in the colored lines, are all driven by these physiological traits, okay, these biological things. How easily does an organism acquire nutrients from seawater? To what extent can it store nutrients above that required for minimum growth, et cetera, et cetera. So these bits of nitty-gritty really matter. So to finish up, before I get buzzed off, I want to say, okay, that's great, but then we want to know about nutrient limitation. So what prospect is there that we might actually better provide large-scale constraints on climate models that might allow us to sort between these models in terms of how realistic they are in their nutrient limitation? So we did this study in the Pacific that was led by Tom Browning at GMR, in this cruise that went across this kind of nitrogen to iron limited region. You can see the changes in the concentration of the tracer. And what Tom did that was fantastic is he conducted these classic bioassay addition experiments and was able to demonstrate this transition from largely nitrogen limitation to iron limitation as we move into the upwelling zone. But then he also took samples for proteomics, so we were able to calibrate these changes from the bioassays against protein biomarkers for nitrogen stress or iron stress, which was really cool. And what was even cooler was that Tom did something unusual, and he took a radiometer on the ship, okay? And he was able to measure from the ship differences in the chlorophyll normalized natural fluorescence of the seawater, okay? And that's in the black line. And what he saw was that there was this big change in the chlorophyll normalized natural fluorescence as we moved into this iron limited region. Okay, threefold more, or even fourfold more natural fluorescence when you're under iron limitation. And then we compared this to the measurement of the same property from the satellite, okay, and that's the red line. And you can see the satellite is able to broadly capture this shift in the biooptical properties of seawater as the system transitions from nitrogen limitation to iron limitation. This is really cool because then we can exploit the entire satellite record and use El Nino as a kind of natural experiment to look at how iron limitation and nitrogen limitation might change in the region over recent decades. So the upper panel is showing you changes in SST in black and chlorophyll. Okay, nothing that exciting there. Close correlation between reductions in chlorophyll during positive SST anomalies. But now you've got this new iron limitation proxy, okay? Suddenly we're able to generate a multi-decadal data set at a very large scale on iron limitation. What we see is that iron limitation increases during the El Nino years, during the positive SST anomalies, But interestingly, we don't see any decline in iron limitation at the highest SST anomalies. So even when the upwelling slows down to its like maximal rate, we don't see actually much evidence of this transition to nitrogen limitation. So perhaps that means that our family of model solutions that generated this shift to nitrogen limitation aren't consistent with this data. And maybe we should be more confident about the models that have more resilient iron limitation in the system, which of course then has implications for the productivity trends that we might expect to see in the future. I want to end by going back to this appalling graphical representation of a nice paper and sort of revisit things a little bit. And the first thing I think is that we need to think about shifting patterns of nutrient limitation in the ocean. And we need to understand what regulates changes in nutrient limitation, how that's linked to phytoplankton physiological traits, and how that then affects ocean fertility. And I gave you an example of shifts between iron and nitrogen in the tropical regions. We also, and I think hopefully this will lead on to Nick's presentation, we need to extend our sort of scope here, and we need to also consider other nutrients and what their role might be in this context. And iron and manganese in the Southern Ocean is a good example. But again, I'd come back to this. Ultimately, they're all controlled by these physiological traits. Now, we need to learn more about these. That's challenging because when we measure traits in organisms, they're organism specific. The models, even the most complex models, we still aggregate quite coarse-grained representations of phytoplankton functional groups. But there is a real link here. Like I said, there is this kind of cascade between what you do about iron uptake up to how well you can project or predict the change in the equatorial Pacific tuna fishery, right? So this is important, all you guys who work on that. Moving forward, I think there's a way of kind of linking between experimental observations that generate mechanistic insight. Okay, we then use ships and other kinds of platforms that allow us to map and upscale. Maybe in doing that, we can generate these kind of proxies for the key processes that we're interested in. And they allow us to generate time series. Either we use these proxies in terms of satellite records, that's the example I showed you. We've also been working with colleagues in South Africa where we generated these proxies for iron limitation that could exploit all historical CCD profiles in the Southern Ocean. And in doing that, we can generate these kind of constraints on actual model behavior, right? The ingredients that drive the engine of the model. In that context, I think we need to think about how can we exploit longer-term records, glacial interglacial cycles, Heinrich events, the Holocene climate record. I think we can't just ignore these because they were happening over different time scales and these kind of things. We need to bring all the knowledge we have, I think, to bear on this problem. And I'm going to close, but I'm going to give you a few questions that I couldn't really deal with. To what extent can we observe these patterns of nutrient limitation in the ocean? What nutrients should we consider? How do we isolate these key traits or groups? Can we use mechanistic insight to sort amongst climate models? And then ultimately, how's climate change going to affect the resilience of key biogeochemical functions, carbon export, silica cycling, calcium carbonate, alkalinity, et cetera, et cetera. With those thoughts, I'll thank you for your attention. And I managed to beat the clock as well.
So, if the buzzer didn't go off, we can take some questions.
Hi, Bethany Jenkins, University of Rhode Island. Thanks for a really interesting talk, Al. Just a question. In the data slide where you're showing that switch from the nitrate limitation to the iron limitation, it looked to me that the iron concentration was pretty constant across that x-axis, and I'm wondering if it's really going into, kind of there's some deviations in there in the iron. So would you consider all of that area that's not nitrate limited to just be primary iron limitation?
So you're asking about
Yeah.
area here?
Yeah.
Yeah, I think that's what we saw. I mean, in this side, it's possible there's co-limitation going on, right? If you look at the bioassay experiments, you see the red, there's, that's basically, the green is adding everything, right? So that's kind of the target. So you can see on this side, you basically have to add iron to get a response. The other side, you get a kind of response from both together, right? There's a kind of additive response. And the proteins, we don't find any of the nitrogen stress biomarkers on the sort of left-hand side of the transcript, for want of a better word. But you know, it's a subtle point that they all seem to be under stress in the gyre, right, where the iron is very low.
Okay, thank you.
Thanks.
Hi, I'm over here. Thank you for a really great talk. So I'm a little bit confused, and my buddy, who I was supposed to ask,
Let you down?
No, it didn't.
It's not very reliable.
So maybe you answered it with Bethany's question. Why do nitrogen and iron limitations switch in a global change scenario? That's the one question. The second one is, we get more DIC into the water. Does that at all change things?
Okay, so whether or not nitrogen and iron limitations switch really depends on the trait assumptions that we made, right? So in one of our experiments, we saw no change. Go back, right, on the right-hand side here, iron limitation in red, basically stayed constant more or less over the entire experiment. In this one, we did see a change, right? And that's because, you know, because the iron limitation not as strong, it's still limiting, right? So this is where we get into a little bit of a nitty-gritty distinction in the models, right? The models will assign something as limiting only if it reduces the growth rate more than the other thing. Does that make sense? So iron is limiting on the right-hand side, but it's not as strongly limiting. Iron concentrations are slightly higher because iron is being retained by biology, okay? So it can very easily, once you start to change the climate, you reduce the upwelling, you reduce the nitrogen supply, you can very quickly bring the nitrogen-limited growth rate down below the iron-limited growth rate, okay? And this is this abrupt switch that is a facet of our models, and it's a real problem because, as you can imagine, anytime you have a step function in kind of model behavior, there's a potential for real uncertainty about where does that occur, okay? Hopefully that answers the first part. DIC, do you mean in a sense of carbon being limiting to phytoplankton or about the acidification side?
In areas where there may be sufficient nutrients, but you may run into then bicarbonate limitation.
I see. I think in the model, we don't see this, right? We assume effectively optimal CCMs, right, that they've got these carbon concentrating mechanisms. So the speciation of the inorganic carbon doesn't, you know, we don't consider carbon as a nutrient. Now, that doesn't mean that there can't be situations, you know, in very stratified systems potentially where the carbon speciation changes that you need to use bicarbonate or something like that. But I don't think, you know, on land, CO2 fertilization is a real issue, right? So that's absolutely happening. In the ocean, I don't think we're seeing a fertilization from adding extra DIC. Hope that answered the question.
Yeah. Thank you,
right.
Alessandro.
You get me up. Okay.
And I would like to introduce our next speaker, Nick Harko from University of Hawaii. And I'll turn it over to you.
Yeah, cool. Well, I appreciate the invitation to speak. This is a real honor. So I was a joint program student not so long ago, and I used to listen to the live stream from the lab when I was supposed to be analyzing lots of samples and then sneak into the tent and steal some lobster. So it's nice to be here officially and to be able to contribute as opposed to just taking and taking. So, right. Well, you're going to see in a second that I'm going to be talking about these other elements that Alessandro alluded to. These are, I think, things that sometimes people only focus on when they're making phytoplankton media and trying to get things to grow. And so the real question of this talk and what I want to talk about later on in the week is if these are actually limiting nutrients and what we need to do and what measurements we need to make in order to be confident about those assessments. And so I wanted to, you know, it's Cape Cod in the summer, and so I wanted to start with JAWS. Just when you thought you were safe. And really, like, if I can distill my talk, or if I don't finish on time, you know, there's a sort of visual metaphor here about, you know, what happens when sharks lose their front teeth, right? They have this layers right behind them to take their place. And I think that's how we can think about these extra nutrients, cobalt, manganese, zinc, these other elements that can, I think, limit productivity under certain scenarios. Cool. So yeah, so if you go to cruise, if you go to Station Aloha and you love it so much that you want to take part of Station Aloha home with you, you need to take some seawater and you need to add stuff to it. You can't grow Philocratis at home without supplementing it with nutrients. Some of these are obvious: nitrogen, phosphorus, but some of these, iron, obviously, but then some of these are less obvious. We need to add cobalt, we need to add zinc, probably manganese, some other elements too, nickel, selenium. And so the question is this just an artifact of period in time where we didn't know what phytoplankton needed, so these were thrown in, or are these playing some essential role in affecting marine ecosystems? That's why they need to stay in these media. So Alessandro alluded to this already, but we can take this sort of history of nutrient amendment experiments and plot these like in this nice synthesis here, and we can divide the ocean up into basically two groups, right? There's the subtropical ocean that's largely limited by nitrogen. Those are the blue circles here, right? They respond to excess nitrogen additions. And then there's the iron-limited ocean, the Southern Ocean, and in upwelling regions. Those are the red circles there. And this is a really nice conceptual model. It's easy to teach the students. We're done with it in a second. But you can see that there's a lot of sort of other colors here on the fringes, these secondary effects. Look at all these other elements that come into play. Maybe you thought about silica limitation of diatoms. But I would say there's a good amount of evidence showing B12 limitation, zinc limitation, cobalt limitation. Here's one of my favorite examples. Here, this is from Tom Browning's work in the South Atlantic. And there, they were in the oligotrophic South Atlantic, low chlorophyll. But they add nitrogen, almost no response. They add iron, and there's almost no response. They add both of them together, and that's when you see some response. So co-limitation, you add both, you release the control on biomass, things can grow, chlorophyll increases. And then in this case, you add cobalt or B12, you get even more growth. This is something that's been demonstrated only a handful of times. So the question is, how can we take an oceanographic perspective and start applying this to scale? Okay. So I was first taught this resource ratio theory from Mick Follows and from some other people. This is a nice sort of unifying concept that we can investigate. How much of a given nutrient do plankton actually need to grow? Do we need to sustain the ecosystem? And this is a really elegant approach. It just takes two equations. One is just a Mannino growth equation. This is a growth as a function of a nutrient concentration, which we can get from culture data. And another is a simple steady state ecosystem approximation that we can say that growth is balanced by mortality and export, some kind of conservation of mass. What we can do is we can rearrange these so we have the resource concentration here, R star, that supports the growth of phytoplankton at a certain level. And the idea here is that if the resource concentration, if nitrogen is at its R star concentration, then nitrogen is limiting, and if the R star is higher than what the nitrogen concentration is, then those species are functionally excluded from the ecosystem. They can't grow, they can't compete, they're not going to be observed in that biome. You can think of this as sort of a sticky concentration that would be found associated with a specific ecosystem. To understand if it's limited by nitrogen or iron, this would be another way to diagnose nutrient limitation. Just looking at this nice review, you can see as you move from an iron-limited ecosystem where iron is very low, you start adding iron in, you start triggering this transition, nitrogen limitation. Then if you keep adding iron, you start to stimulate nitrogen fixers, and you ultimately end up phosphorus limitation. And the abundance of these nutrients are sort of shifting in concert here. So we can use this pretty well to explain patterns of phosphorus that we see around the world. So across the gyres, even though nitrogen is very low in the North Atlantic and the South Atlantic, there are big shifts in phosphorus concentration that we can start to disentangle and associate to, let's say, iron supply from the Saharan dust. So that unlocks nitrogen fixation, and that leads to an uptake phosphorus down to that R star level. So that's demonstrated pretty nice in this Moore 2009 paper. But also worldwide here, you can see that anywhere where you have high dust off of Australia, off of Asia, in the Mediterranean, you see the same thing, this trend toward lower phosphorus. This theory holds up, and so what I want to do is connect it to these other ecosystems here. So we have this basic concept of iron limitation transition to nitrogen limitation to phosphorus limitation. But what about these other metals? So what I want to propose is that there are these intermediate checkpoints offered by manganese, by silicate, by zinc, and by cobalt that basically are different stops on this ecosystem that can stick here at these R stars if there's not enough cobalt or manganese around in this ecosystem. Okay, and then obviously there's various physiological adaptations, these different traits, some of which that Alessandro mentioned that complicate these things. We're not going to stick around these too much, but these are ways that these different nutrient conditions can interact with one another and increase zinc requirements under phosphorus limitation, for instance. Okay. So the first element I want to deal with is cobalt. Cobalt is my favorite element, just for conflict of interest purposes. And so if you don't know about cobalt, it's fine. It's not a very hard biochemical cycle when you boil it down. There's essentially two key parts. One is cobalt 2+, the metal that's taken up by organisms like Prochlorococcus. It's turned into a B12 analog that's used to power certain enzymes to make DNA, to make proteins. That's one use of cobalt for a primary producer. Some of that B12 is recycled and available to eukaryotes, who use it for very similar purposes. And there's a second function here where cobalt is used to replace zinc, to fulfill this carbonic anhydrase, zinc finger proteins, and other sort of biochemical uses of zinc if zinc isn't available. That's sort of the cobalt cycle in most ecosystems. And so based on some experimental data with Prochlorococcus that we did in the lab, we can start to say, well, how much cobalt does this ecosystem need in order to do those functions, to provide enough B12 for everything else to grow? And so we can calculate this R star just by putting some dummy parameters in here, taking a value extrapolated from culture experiments like this, where we can grow Prochlorococcus, we can change the cobalt concentration, and then see how the cells respond and estimate where that tipping point actually is. And if we do that, we get something like 1 picomole of bioavailable cobalt. It's a really small number. Can't actually measure bioavailable cobalt directly, unfortunately, so we have to add in this fudge factor of this bioavailability factor, how much bioavailable in the total cobalt pool. That's a made-up number. Some information goes into this, but let's say 5 picomolar level is what the R star concentration would be. And so what we can do now is go and look at the CO traces atlas, the intermediate data product, and we can see, where in the world is cobalt at this R star concentration or below it, or is it, is it replete everywhere? That's what's shown in this map. There's a histogram here, but you can see that there's some major parts of the ocean where cobalt is very close to this threshold. This is not cobalt limitation per se, but these are definitely where it's sort of concerning, or at least should be on your mind. And it covers big parts of the South Atlantic, especially the subpolar region, and also in the South Pacific here. Large parts of the ocean don't have a lot of cobalt. It's far from land, far from the closest source. Cobalt's low. Maybe around 6% of the samples that we have observed, the traces there fall below that threshold. So let's dig into this a little bit more. This is work that I did as part of my PhD in Max Beto's lab. This is from this transect, this Metzyme cruise from Hawaii to Samoa. And this is the distribution of cobalt here in this middle panel. You can see there's high concentrations 10 north and 5 south. These line up with low oxygen zones. We think there's a big cobalt source with low oxygen regions that makes its way into the Central Pacific. That's upwell to the surface and then spread out into the open ocean. But by the time you get to 10 south in the South Pacific Gyre, you have water with no cobalt left in it. So these are these areas where you're below that R star threshold where you say, maybe there's cobalt limitation here. And just to give you an example of what the profile looks like, you can see that throughout the upper 400 meters of the water column, cobalt and phosphate are tracking one another. And then once you get to the photic zone, concentrations go to 0. You might say, well, maybe there's organisms here that don't need cobalt to grow, but we know from pigments that there's divinyl chlorophyll A, and that has to be made by Prochlorococcus. There has to be a lot of cobalt requirements here. And if you look at what ends up in the particles, particulate cobalt here in pink tracks the particulate phosphorus, which we think is associated with biomass. Things are taking up cobalt. It must be recycling it at a great rate in order to provide growth for this Prochlorococcus. We have another constraint on how much cobalt it might be needed to power this ecosystem based on the particles. And we can go back to our culture experiments with Prochlorococcus to basically estimate, based on how much they've accumulated from their environment, what do they need to grow? This is a droop type model, so we can say the particles have 2 picomoles of cobalt, they have 20 nanomoles of phosphate. Where does that track on limitation experiments where we're actually able to confirm cobalt limitation? And we can assign certain growth rates here and say this is a range of cobalt to phosphorus ratios we associate with limitation, big growth, with ability to grow Prochlorococcus. And so we can connect this to the field data again. So here is the dissolved phase cobalt measurement. So you can think about this as if Prochlorococcus is responsible for taking up all the residual phosphorus in this ecosystem, it's going to have to grow at a cobalt limited rate in order to do so. right? We don't have enough cobalt relative to phosphorus in this ecosystem to support rooflet growth, so we can exhaust things there. But somewhat counterintuitively, if you look at the particles, there's actually plenty of cobalt there to satisfy the daily growth rate that you might expect for the prochlorococcus. So we're in this sort of middle ground here where the cells are not going to be instantaneously cobalt limited, but at some point, if we're to keep adding iron or other limiting nutrients to this ecosystem, they would run out of cobalt. We can look at this graphically here. So I've plotted a cobalt to phosphorus ratio on the y-axis here and iron to phosphorus ratio on the x-axis. You can basically divide up the ocean into these different domains here. So in the top corner, there's plenty of iron, there's plenty of cobalt. You expect nitrogen or phosphorus limitation to be dominant here. But over here, we've got cobalt limitation thriving because we have high iron supply and low cobalt supply. And over here in this quadrant, there's plenty of cobalt, but there's low iron. So we can see that most of the observations across the ocean, as shown in these X's, would fall across this N to P limitation regime and then this iron limitation regime. And it's only once you get into the South Pacific, we get very close to this line of equal limitation. So what does this mean? It means that if we were to add a little bit more iron, we would transition this ecosystem to something that would be cobalt stressed. So we can think about the South Pacific today as something that is not cobalt limited right now, probably, but could easily become that in the future, and would not sort of lead to this phosphorus limitation that we'd expect from the resource ratio paradigm. All right, that's one side of this cobalt story here, but there's a second usage as a substitute for zinc. And I want to briefly sort of cover this as case study 1.5. We can do the same approach that we did before. We can take representative zinc growth data from culture. We can apply trustee bioavailability factor, which is, again, made up, but somewhat supported. And we can say, well, where is zinc low? And we can say it's also low in parts of the South Pacific, also in the North Atlantic, also in the South Atlantic. So there's large parts of the oligotrophic ocean that are low in zinc. We might be concerned about zinc limitation as well. And there's some really nice work that recently came out from Riskel and Max Lab. And what they did was basically grow diatoms under zinc and cobalt co-limitation. You grow diatoms without zinc, then they take up cobalt and use it the same way. But in either case, if you deplete both elements, there's these two proteins that turn on, these zinc cobalt responsive proteins, they named them that. And they turn on in basically the sort of low picomolar to sub-picomolar range. And across this mezyme transect, these proteins turn on, they're abundant in the South Pacific gyre. The second indicator that we're in the stress zone in the South Pacific where we're both depleted in cobalt and zinc. Okay, so we can basically just modify our plot before. This is a cobalt or zinc co-requirement for Emiliana-Huxley eye. You can see it's higher than cobalt. They need more cobalt here because it's substitute for these zinc roles and carbonic anhydrase and other enzymes. And we can see there's this larger swath here in this shaded region where we're in the stress zone, or we expect to be in the stress zone. That covers a lot of observations throughout the Southern Pacific especially. So now we have two elements that are going to be limiting before phosphorus. So the last case I want to talk about is manganese in the Southern Ocean. So again, we can do this R-star approximation, say how much manganese does the ecosystem need to survive using our same sort of representative growth rates, and also some a factor derived from culture experiments that we have. This leads us to 166 picomolar, and we can, the nice thing about manganese is there's very little bioavailability uncertainty that we have to play here. We can just describe that number literally. And across this geotraces data set, you can see in the histogram there's these sort of two major populations, manganese concentrations across the world. There's one around 2 nanomolar or so. This is tropics, subtropics, everywhere else. Plenty of manganese, not a worry. But then when you get down to 0.1 nanomolar, or yeah, 0.1 nanomolar, that's pretty close to this R star, and you have this whole population of measurements that shows up. These are all in the Southern Ocean. So what's happening here is manganese is a scavenged element. It's a low inventory in deep water. And as soon as you start upwelling deep water in the Southern Ocean, you're giving an ecosystem with lots of nutrients and no manganese to grow. So this is not only a stoichiometric argument, there's also some bottle incubations that were done recently in Drake Passage that show you take water from the Drake Passage, you can add manganese and see growth after a couple of days. If you add iron, you also get a response. And if you add iron and manganese together, you get a bigger response overall. This is also a co-limitation that seems to be impacting large parts of the Southern Ocean. Really could be. So one of the things that we were working on, this is with Alessandro and Ben Twining, try to scale up the observations that we had, try to account for as many processes that we could think of, because the manganese is being used in photosynthesis. That's dependent on light. So the ocean is very dark in the winter, kind of light in the summer. There's lots of photoacclimation going on. There's changes in growth rate. There's temperature going involved. So we wanted to try to account for as many of these processes as we could. This is the sort of manganese distribution in the model. You can see it's mostly low, except for coastal areas. And we try to sort of take in the known physiology here. So the other thing besides this photosynthetic usage, which determines the manganese quota, how much the cells need to grow, is its interaction with these other metals. So unfortunately, manganese has a very low ability to compete with other metals for phytoplankton transporters. So whenever there's any zinc around or any copper, then that basically blocks the uptake of manganese into the cell. So the notion that's a big deal because you're upwelling water with high zinc. So the model turned out that because there was significant manganese limitation at various points throughout the year, anywhere where this sort of turns yellow is areas where manganese limitation seemed likely. If you compare it to iron limitation, it's not that impressive. It's only a couple percent. Total ocean is more manganese limited than iron limited. It's not going to overturn iron limitation experiments that have been demonstrated throughout the last several decades. But there are seasonal phasings here, and so if you account for the change in carbon export, there is a pretty large difference in certain regions due to current day manganese limitation. That's because of the seasonal phasing where we think manganese limitation is more intense in spring. So iron limitation is more intense in summer. Okay. But there's sort of a bigger problem where, Alessandro mentioned this, is that in these models, the limitation is assessed at the current growth rate. So if you're heavily iron limited, most times your manganese requirement is also going to be low because the cells aren't growing very fast. They don't need to take up a lot from their environment. If you were to hypothetically add iron, you would immediately confront the ceiling of manganese. So that's what this manganese efficiency plot is attempting to show, is a much larger plot, a much larger part of the ocean is really sensitive to manganese as soon as you give it iron. This is something that we were able to demonstrate in the paper. If you take dust coming in, dust has a high ratio of iron relative to manganese, relative to their biological requirements, and so a last glacial maximum period was probably more impacted by manganese limitation by iron limitation. So what I hope I've shown you at this point is that there's all these interactions between the trace metals, cobalt, zinc, and manganese that are sort of just below the surface of our present day iron and nitrogen limited world. And any kind of perturbation that we embark on, whether that's future climate change, past scenarios, could easily sort of tip things over to be primarily limited by cobalt or zinc or manganese. There's a lot of sort of experimental challenges that we would need to do to model this better. We have a pretty poor cultural representation of. I have 55 more minutes now. But we need experiments with relevant organisms because a lot of that manganese modeling, for instance, came from Glaciosira that was isolated from Long Island Sound. And we're using it because we don't have anything better to do, project it across the entire Southern Ocean. These are data gaps that I think need to be addressed, occur in lots of other areas too. So thanks.
Okay, thank you, Nick. We're going to move on to our first set of lightning talks. And Randy's going to be the first speaker.
All right, I'm going to start us off. So I thought I could offer a little bit of a different perspective on this kind of ocean metabolism and fertility that we're talking about in this session, and on the more kind of dark side of the biological pump, so thinking about heterotrophic bacteria. And this is work that was funded by the Simons Foundation for an early career grant that primarily my postdoc, Travis Mellett, has done, with also a lot of help from graduate student Patrick as well. And the kind of motivation of this project is that if we look below the euphotic zone at profiles of dissolved iron in seawater, which are in those blue dots, and then a profile of nitrate, for example, in orange, they have a little bit different shapes. And so in the mesopelagic ocean, we actually have pretty iron deficient conditions, similar to the surface waters, and it doesn't really increase until this kind of much deeper regeneration length scale compared to macronutrients. So we've been thinking a little bit about whether or not these low iron conditions are affecting heterotrophic bacteria because they also need a lot of iron, for example. And we've seen some other pieces of evidence and other types of data to suggest that perhaps heterotrophic bacteria in this zone might be a little bit iron deficient. For example, we see a lot of siderophores, which are compounds produced by heterotrophic bacteria to facilitate iron acquisition from the environment at these particular depths. So it gives us kind of a proxy that they might be at least struggling to get enough iron. And so we wanted to ask the question if heterotrophic bacteria in the mesopelagic specifically are iron limited. And so we've done 4 experiments in the kind of general North Pacific, which I've highlighted in that map up there, at the equator and also in the subtropics. And we've completed these experiments at different depths within the mesopelagic zone. And this is just one example here where we gave various different iron and carbon additions, incubated them in the dark, and looked at various growth properties. And so this is showing the 3 depths from one experiment, starting at 200 meters, going to 450 meters, going from control and then adding just iron or glucose or a combination of the two. And basically, the summary that we saw in all the experiments is that we got higher growth rates and higher heterotrophic bacterial biomass when iron and carbon were added. And this, in some cases, was surprising because even when we added really labile glucose at a lot of these depths, nothing happened. So it's kind of pointing to this co-limitation between the two, like Nick was talking about, which is not necessarily what we would expect with heterotrophic bacteria. We kind of think of them as always carbon limited almost. So this was already kind of interesting. And the other parts of it that we thought were interesting is that when we gave them more iron, there was higher bacterial growth efficiency when it was present, and there was also an enhanced total carbon consumption when we gave them iron as well. And interestingly, in experiments where we just gave them carbon, we saw this kind of like outsized siderophore production. So they kind of turned it on when they still had sufficient carbon, but not quite enough iron. So that could be explaining why we sometimes see these kind of siderophore maxima in the mesopelagic as well. And it suggests that they're kind of dedicating energy towards iron acquisition. And then they had very, very high iron quotas. So we look at that as kind of this iron to carbon ratio, and they showed a lot of plasticity in our experiments, ranging from greater than 1,500 to around 7, which is what has been observed before. And this is quite high if we compare it to, say, like a diatom iron to carbon ratio, which is around 2. So they have this like big range. And so what we kind of think is that mesopelagic bacteria have these like pretty high iron quotas because they're just kind of storing iron until they get some sort of pulse of carbon. Then they can use that iron and kind of churn through that carbon, and it really kind of is impacting how fast they chew through that carbon, how much of it, and how efficiently they do. So we haven't connected it quite to this kind of like ultimate goal of the effect on the bacterial carbon pump, or the biological carbon pump, but we think it's something that we need to be thinking about, especially with kind of iron fertilization experiments, all these different things, and the efficiency of carbon export. All right, and that's it. Thank you.
Hello. Hello, my name is Ichiko Sugiyama, and I'm currently a postdoc at CUNY. I work with Mac Saito and Tristan Horner. I'm funded by CUNY and OCIA, and I would like to thank everybody who helped me make this work possible. And today I will be talking about microbial metal demand in the Eastern Tropical Pacific assessed using iron and copper isotopes. So last summer, I went to the Eastern Tropical Pacific, where I started the cruise, AT50-10 from Costa Rica, partied at the equator, and then ventured off to HNLC, low iron at the surface, went through oxygen minimum zone, and finally ended at San Diego. And the objective of today's talk is to show you some of the results from one of my projects where I determined the microbial iron copper demand in the Eastern Tropical Pacific, ocean profile, and ocean transect. So how did I do this? So we developed a stable isotope metal uptake method where step 1, get trace metal clean seawater. I sampled 12 to 18 depth profile because who needs sleep? And I collected all those seawater and I spiked with iron 57, so this is an enriched spike, and copper 65 iron 57 at 1 nanomolar concentration, and copper 65 at 0.1 nanomolar concentration. I then capped, shook the bottle, and incubated for 24 hours by varying light intensity, diurnal cycle, and temperature. I then, after I finished the experiment, I filtered the sample, stored it until I digested the filters and measured the metal concentration using the ICP-MS. So before I move on to showing some of the results, I would like to tell you how I plotted my results. So all the metal uptake results are reported as isotopic ratio. So let me explain how this works. Example I'm giving you here is copper, where copper 65 spike was added as 0.1 nanomolar, where the spike copper 65-63 ratio in the x-axis here, plus somewhere on the right side of the figure here, and the natural abundance, the background, essentially plots on this gray dotted line. So any data point that plots towards the cat indicates that there was copper uptake, and same for iron. So remember that. So let's look at the results. So copper demand in the Eastern Tropical Pacific from Station 2 to Station 5. Immediately here, you can see that there's a variability in copper uptake over ocean profile and ocean transect. And you see uptake of copper at the surface, but also in the mesopelagic. That's interesting. And same thing for iron. There's an iron uptake variability over ocean profile and ocean transect. Again, there's iron uptake at the surface and in the mesopelagic. And this uptake at the surface makes sense, but what's going on in the mesopelagic? So one of the hypotheses we have is that potentially there may be a microbial metal demand in the mesopelagic. And looking at the nitrogen cycle, many of the metalloenzyme in marine microbes require iron or copper. And looking at the data published by Santoro et al. 2016 paper and Saito et al. 2020 paper, where they quantify the abundance of metalloenzyme over ocean profile from methzyme and proteome cruise, you can see that these metalloenzymes that require iron and copper have high abundance in the mesopelagic. So the take-home message here is that metal demand varies over ocean profile and ocean transect, and we see iron and copper demand both at the surface and in the mesopelagic, and there may be a potential for microbial metal demand in the mesopelagic. And stay tuned for more results because I have 11 more stations to go. And yeah, a lot of data. And also we're starting to measure the proteomics data as well as getting results for POC, particulate metal and seawater as well. So stay tuned for more results. And if you want to know more about the uptake rate, I also calculated it. So come to my poster tomorrow. Thank you very much.
All right. Hi, I'm Drew Chappell, and I am going to talk to you about interactions between iron, light, and silica limitation at the subantarctic front. I'd like to acknowledge my funding from NSF, and I'd also like to acknowledge the overall scale Southern Ocean seasonal experiment. We were able to join one of their cruises, and they're doing some regular cruises between South Africa and the Antarctic, crossing all the wonderful fronts. So what we set out to do was to look at iron and light potential for limitation or co-limitation in two different incubations. I'm going to mainly, I have both of them on my poster, so plug there for tomorrow. But I'm going to talk to you about what we saw at the subantarctic front. So we did these controlled incubations with 5 treatments: control, medium, light, no iron added or removed. We had 2 high iron treatments where we added iron. We had 2 low iron treatments where we removed iron, sort of by chelating with desferoxamine B. And then we had high light and low light. We did these in these very cool incubators by Sven Kranz that have a LED light array that does light like you see light happening where it goes up and it goes down, and 12 different wavelengths of light. And these are the averages and max light for the different treatments. So on the left, I have the growth data that was done by chlorophyll measurements throughout the incubations. And what we see is a really nice response where we have both a growth response when you add iron versus the iron removed or versus the control, and then a slight growth increase with higher light over lower light. On the right, I'm showing you some of the molecular data. So we specifically, the metatranscriptome reads that were classified as diatoms. We're looking at the differential expression from the different treatments, and I apologize, the colors are not coordinated on that slide. A little bit of a fail of R. But what we have is sort of the different treatments, so all of the individual replicates of the different treatments and how similar the gene expression profile for the diatoms genes were in the different treatments. And what's beautiful to see is that they group together by treatment, thankfully. And we see that on the PC1, so the first principal component, which explains a lot of the variance, that is high iron on the right versus low iron. And then the PC2 is basically separating out the low light and the high light. So all the replicates are grouped by treatment, and we're seeing an iron impact. So then we wanted to know a little bit about what we're seeing in those genes, and I've only started this analysis, so this is the teaser of what I've been seeing. So what this is, and it is looking at the genes that were upregulated under low iron on the left versus high iron on the right, and these are the protein family functions that were enriched and upregulated in low iron. And if you've seen some, unfortunately I'm not after Harriet, who will talk to you some about metatranscriptomics, probably not with iron, but flavodoxin is something that we regularly look for as an iron stress marker, and that is, so typical iron stress marker genes are upregulated and enriched when iron is removed, so that was really heartening to see. And then on the right, what we saw, the function that was most enriched once we added iron back in were the silicon transporters, which was really confusing to me originally, but it suggests that we've reached a point where the cells are experiencing silica limitation. And so basically we pushed them to silica limitation, which makes sense because where we were at the subantarctic front, the nitrate is still really, really high, but the silica has started to drop off. So it was below 1:1. So thank you.
Hi, everyone. My name is Hope Elliott. I'm a fifth-year PhD student at the University of Miami, and today I'm going to be talking about volcanic ash and its impacts on ocean biogeochemistry. So a lot of studies have proposed that volcanic ash fertilizes the surface ocean. Essentially, you have an eruption, some amount of ash falls into the ocean, and presumably nutrients leach out that then are bioavailable and stimulate a phytoplankton bloom. And most studies focus on one nutrient specifically, iron. But what's interesting is that there's not much evidence that most or even many eruptions actually drastically increase dissolved iron concentrations, and different leaching experiments have provided very different estimates for the amount of soluble iron in volcanic material. So we wanted to look into this. We collected ash from two different eruptions that occurred on opposite sides of the Atlantic in 2021, the La Soufre eruption in April and the Cumbre Vieja eruption in September. And we measured soluble and total iron content in ash from both of these eruptions. We had 3 samples from Cumbre Vieja and 1 from La Soufre. We ultimately found that the Cumbre Vieja ash contains more total iron and more soluble iron, and this also results in a higher % iron solubility for ash from that eruption. So on average, iron in volcanic material from Cumbre Vieja is about 4% soluble. Iron in the La Soufre ash is only about 1% soluble. So what does this then mean in terms of ocean fertilization? So we also looked into particle size, and we found that the La Soufre particles are generally much smaller. They have a sub-micron mode diameter. The Cumbre Vieja particles are much larger. That means they're going to be deposited closer to the source. It also should be noted that both eruptions occurred in locations where the background near-surface dissolved iron concentrations are generally less than 0.5 nanomolar. So we then use the particle size, the soluble iron content, and the eruption length to estimate increases in dissolved iron concentrations as a result of both eruptions. So based on our calculations, the La Soufrière eruption would have basically a negligible impact on dissolved iron concentrations in the mixed layer, but the Cumbre Vieja eruption with those larger, more mafic particles could theoretically enhance dissolved iron concentrations and increase them by about 20%. We should also consider some other nutrients. We recently published a study on phosphate in the La Soufrière ash, and unfortunately, kind of like iron from this eruption, there is some phosphorus there. It's not very soluble. We also looked at some of Nick's favorite elements in the Cumbre Vieja ash, and what's interesting is all four of these elements, phosphorus, manganese, zinc, and cobalt, are present in this ash sample at abundances that are above the crustal abundance. So potentially this ash would add more of these nutrients than the major aerosol input to this region, which is mineral dust. The other thing to consider is you also do get some toxins in volcanic material, so potentially you're getting this flux of nutrients, but also toxins to the surface layer. And we ultimately concluded that some eruptions likely have the potential to add biologically relevant fluxes of iron, but the actual biological impact probably depends on the exact composition of the material and whether or not iron or a different nutrient is limiting in the receiving environment. But this is actually not what my poster for this conference is about. My poster for this conference is about something very different. It's about this discrepancy. Why is iron in one type of ash 1% soluble, but it's 4% soluble in a different type of volcanic material? And there's kind of an ongoing debate in the aerosol community about what actually determines nutrient solubility. There are a couple kind of factors people consider. Mineralogy, with iron specifically, iron in things like silicates, for example, tends to be more soluble than iron in oxides. Particle size is also important, so if you have the same amount of material, theoretically more nutrients should be leaching out of a smaller particle due to its larger surface area to volume ratio. And then especially in an acidic eruption plume, you generally get some type of acid processing, which is thought to enhance nutrient solubility. And for volcanic ash specifically, high levels of fluorine have been linked to elevated iron release. So like I said, this is an ongoing debate. People want to know which of those three factors are the dominant control on nutrient solubility in volcanic material. Unfortunately, I don't have time to tell you what we found in our talk. We did do a lot of mineralogical and particle size analysis, so we do have some ideas about this, but if you'd like to know them, please come talk to me at my poster. It's called Volcanic Ash Deposition into the Surface Ocean, Mineralogical Controls on Iron Solubility.
All right, hi everyone. I'm Frankie Pavia. I'm an assistant professor at University of Washington. I'm going to be talking today about some work funded by NSF Chemical Oceanography to couple the biogeochemical cycling of iron to something that has absolutely no biological role, which is thorium, and how we've been improving iron models by doing that. So global iron models, right, generally consistently simulate dissolved iron concentrations. So on the x-axis here, we're looking at different biogeochemical models of iron, and the average model dissolved iron on the y-axis with the observational mean plotted on there. So, you know, within like 50%, they're doing an okay job. But if we look at how they do this, all these models simulate the same concentration of iron with quite different parameters. So on the plot on your left is the total modeled iron flux into the model on a log scale, and on the right is the residence time of iron on a log scale. So they're achieving these same iron distributions with wildly different input and removal rates. So, right, if we want to prognostically model biogeochemical feedbacks in the iron cycle, we need constraints on iron input and removal, not just concentrations. So, right, this is where thorium comes in, and Chris Hayes made this really nice figure. Iron, much more complicated. Thorium, simple, right? Thorium is like the caveman of the metals. Right, so iron has biological effects, it's scavenged, it has redox, right? Like all these sorts of things are affecting it. Thorium-230 is produced in seawater at a constant rate by uranium decay, and it's scavenged by sinking particles. That is its geochemical cycle. There's another isotope of thorium, thorium-232. It comes in by dissolution of aerosol dust, and then it gets scavenged onto sinking particles and removed. So we actually have some tools here that can give us constraints on aspects of this complicated iron cycle with less of the sort of complicating factors. Right? And so in recent years, we've been finally, thanks to geotraces, starting to get enough globally distributed thorium data to be able to model in a global biogeochemical model. So what I've been doing with Keith Moore and Weilei Wang is putting dissolved thorium isotope model into the marble, into CESM's biogeochemical model. And so, right, the way that we did this at first was just say, all right, let's model thorium exactly the way that we've modeled iron with only the scavenging term for thorium-230, right? And here's how we did. So, right, the model is plotted on the y-axis and the observations are on the x-axis. So we can see that in the mid-depth water column, the sort of center of this plot, the model seems to be doing okay. But at the surface ocean, down in low thorium concentrations, and in the deep ocean, up in high thorium concentrations, the model is overestimating the concentrations of thorium. So the scavenging model that was being used for the iron was not doing a good job of representing the dissolved thorium in the model. And so we developed a new scavenging model where we basically cranked up the rate of scavenging in the surface ocean and added a bottom scavenging parameterization as well. And this let us fit the thorium isotope distributions with the model quite well. So this is the radiogenic isotope of thorium, the one produced at a constant rate in seawater by uranium decay. This is thorium-232. So this is the one that's brought into the ocean by the deposition of aerosol dust. And you can see that the fit is a little bit worse here, but we were able to do something neat, right? Which is that some places here, if we go back to thorium-230, there's still biases in the model compared to observations based on still inaccurate scavenging parameterizations. And so we went ahead and corrected the thorium-232 in the model for those scavenging biases at each of these points and looked at what we had, right? And so for parts of the ocean, the model is doing a pretty good job. But for parts of the ocean, in particular the South Pacific, down at the bottom left of this model, and the Southern Ocean in orange, our model is really underestimating the amount of crustal thorium-232 that's getting to these parts of the ocean. And right, we've already corrected for scavenging bias in the model. So this is an inaccuracy in the dust deposition in CESM. And this is comparable to what we've seen in observations where we've taken thorium isotope profiles and converted them to dust fluxes. That's shown on the x-axis. And the community atmosphere model's dust flux is shown on the y-axis. And we can see that these models are consistently underestimating dust fluxes in the southern hemisphere in low dust flux regions by orders of magnitude. Okay, so where does this all leave us? I want to leave you with this takeaway here where we're showing the model to data fit in the old iron model before our scavenging improvements on the left and our new iron model on the right. And we're doing a better job of fitting the observed iron concentrations. And the places where we see deviations are quite tantalizingly, I think, in, say, the South Pacific and in the Southern Ocean, where we think that we can improve by improving the atmospheric deposition rates. So I'll leave you with that. Thank you.
All right, and we are going to be moving right into our next set of longer talks. All right, and our first talk after the first round of lightning talks is going to be Rob Letcher from the University of New Hampshire. Take it away.
All right, thank you. Today I'm going to be talking to you about organic nutrient fertilization of the ocean's biological carbon pump. Just to start off, I want to point out this acronym BCP, which I'm going to be referring to throughout the talk, is referring to the ocean biological carbon pump. So I want to start my discussion this afternoon by posing a question, and that is, does the ocean's meridional biological carbon pump gradient mirror the meridional gradient and a number of other well-known and established biogeochemical tracers? For example, chlorophyll A, which I'm showing here in the top left. I've also wished to point out the Hawaiian Ocean Time Series site and BAT site, which I'll be referring to throughout today's talk. Also, this meridional gradient you can see here in surface nutrients such as nitrate I'm showing here in the bottom left, as well as net primary productivity. This is an estimate from satellite via the CAFE algorithm. And what you can see, this is a pattern I'm sure is familiar to many people in this room. You see these elevated rates of MPP or chlorophyll in polar, subpolar, and equatorial regions, and much lower values in the subtropics. And this meridional pattern is, we think of as this existing paradigm where the biological productivity in the surface ocean is limited by the delivery of new nutrients from below. And if we look at this last tracer I pulled up here of particulate organic carbon export, and again, we see this meridional, which we think dominates, POC, we think dominates the biological carbon pump strength. Again, we see this meridional gradient, reinforcing this paradigm. And so this meridional, sorry, I should back up. This vertical pattern, where our biological carbon pump is dominated by the delivery of new nutrients from below and export in these vertical processes. This gets encoded in our ocean biogeochemical models. For example, here I'm going to refer to the CESM model, NSF climate model, as well as satellite-based estimates of the biological carbon pump when you couple this with empirical estimates of the E ratio. So what we're looking at here is the meridional gradient in the annual net community production flux, which is equivalent to the biological carbon pump, from Paul Quay's paper from a couple of years ago. And so we have this meridional gradient in the Pacific on the left-hand plots and the North Atlantic on the right-hand plots. And so we see this, this mirror of this image of the meridional gradient I was talking about before, this kind of this high, low, high, low, high meridional gradient in the models, both on the top and the bottom plots here. But what do the data tell us? And so the observations that Paul is showing us here in the black are oxygen and CO2-based geochemical estimates of annual net community production flux. And there's this much reduced meridional variability. All of these estimates vary in the narrow range of about 1 to 3 moles of carbon per meter squared per year. Especially where these models and the geochemical observations kind of diverge most strongly is in these subtropical latitudes, right, where this lower nutrient ecosystems, lower input of nutrients from below, yet we see this kind of maintenance of biological carbon pump strength. And so this begs the question, how is the BCP maintained in these low nutrient ecosystems if they're not getting as many nutrients from below? We've kind of been asking ourselves this question for a very long time, right? We have these time series sites located in the ocean subtropics, hot and bats. Since 1988, I'm showing you here the dissolved inorganic carbon drawdown from the mixed layer, right? We observed this pattern very early on in these time series. This is in the surface mixed layer, the upper 50 meters of the water column. There's this strong drawdown in DIC throughout the summer months that's been linked to the biological carbon pump, not so much the seasonal warming associated air-sea gas exchange. Right, and this is a time of very strong stratification in these systems. Not a lot of nutrients in the surface ocean, and a lot of nutrients are located 100 meters or below from this mixed layer, and so not a lot of vertical processes that we're aware of that can deliver nutrients all the way up to the surface ocean to explain this biological carbon pump strength. So what I've done next is I've tried to create some nutrient demand and supply budgets for these two sites. And so what I've done here, I'm showing you here hot on the top and bats on the bottom. And I've taken that net community production, that DIC drawdown, and converted into a nitrogen demand using the elevated C/N stoichiometry of marine biomass at these sites. So this top number here is showing you the total N demand for these systems, about 350 to 330 millimoles of nitrogen per meter squared per year. And then the very next number down on these tables here is the nitrate supply from below. What you can see is that the nitrate supply is a little bit less than half of the total N demand in these sites. Right, so that kind of gives us already an answer to our question of how the biological carbon pump can be maintained in these ecosystems is that there's other non-vertical processes that are important. Right, and these are the well-known, well-studied processes of marine nitrogen fixation, atmospheric deposition, which we heard a lot about yesterday, and as well as eddy movements. And when we add in these extra non-vertical processes, we get closer to closing, meeting that N demand. The total N sources here about 300 or 250, but we don't close the budget. So what else is important? And this kind of brings me to my main, take-home message from this talk today is that the lateral supply in these systems is important as well. So I'm not only talking about inorganic, so I'm going to be talking today mostly about organic nutrients, but this is also the inorganic nutrient supply. And so this is some of the first estimates of these lateral nutrient fluxes. This is work from Rick Williams and Nick Follows from a couple decades ago now, estimating the lateral supply of nitrate horizontally in the Sargasso Sea in the North Atlantic, approaching 50 to 100 millimoles of nitrogen per meter squared per year, which rival the other terms in that budget I just showed you. And then this model estimate from Torres Valdez here on the right, where they estimate maybe 20 to 40% of the export production or biological carbon pump can be sustained by dissolved organic nitrogen in the Atlantic. And in these systems, right, we are the subtropical ocean has a convergent circulation. It's ringed around by ecosystems that experience upwelling, ultimately producing dissolved organic matter, and these convergent flows deliver this DOM to the gyres. And so I'm going to be talking more today about these, what I'm going to be referring to as organic nutrients. And by organic nutrients, I'm referring to the semi-labile detrital dissolved organic nitrogen or phosphorus pools that have mean lifetimes of days to years, are thus transported by this strongly lateral flow in the surface Ekman layer. And what is this semi-labile dissolved organic nitrogen phosphorus comprised of? There's been many, I'm not a compositional chemist, but there's been a lot of work over the years trying to figure out what the high molecular weight or some of the more bioavailable dissolved organic nitrogen and phosphorus is made of. Dissolved organic nitrogen, we think, is mostly the semi-labile stuff is made up of amide and an almost 50-50 split of proteinaceous-like material and chitin or amino sugar-like material that are broken down by aminopeptidases and chitinases, respectively. And for dissolved organic phosphorus, we think it's dominated by this class of mono and polyphosphate esters, such as this glucose 6-phosphate molecule that I'm showing you here, which is broken down by alkaline phosphatase, and a smaller contribution from phosphonates with a stronger CP bond broken down by CP lyase. All right, so how are these organic nutrients contributing to the subtropical ocean, right? We've been focusing in here on the hot and the bad site. So I'm showing you some work I did a number of years ago using the community Earth system model, which I've already referenced, where I've optimized dissolved organic nitrogen and phosphorus cycling in the model relative to observations of these pools in the ocean. And so I'm showing you here the uptake of dissolved organic nitrogen in the top left and the uptake of dissolved organic phosphorus that's been produced outside the gyres and then mixed into these gyre regions and then subsequently utilized by the phytoplankton there. And so what you can see is that the dissolved organic nitrogen, this is not working anymore, the dissolved organic nitrogen fluxes are about 20 to 40 millimoles of nitrogen per meter squared per year, which put it on par with the nitrogen fixation fluxes in these ecosystems. Where we can kind of see a little more clearly on the dissolved organic phosphorus plot on the bottom here, that especially along the equatorial flanks and poleward flanks of these gyres is where seemingly a lot of this semi-labile dissolved organic matter is being mixed in. And now what I can do is I can go pull out bats and hot from my model output here and compare the total amount of N demand and phosphorus demand supplied by this dissolved organic nitrogen and phosphorus, which is in these orange bars here, and find that at these time series sites, it's about 12 to 20% of the N demand can be met by this lateral DON supply, and for phosphorus, it's about 20 to maybe up to half of the phosphorus demand in the Sargasso Sea is met by this DOP. So in a model, what are the some field observations tell us? So here I'm zooming in on dissolved organic phosphorus and some recent work on an NSF grant that myself and Angie Knapp at FSU have been working on, where we really tried to augment the observational coverage of dissolved organic phosphorus. Previously had been focusing on the Atlantic basin for DOP and the black dots, and we added in all of these red dots here of dissolved organic phosphorus. And what the project PhD student Zhou Liang at FSU did was plotted these upper ocean DOP stocks along these meridional zonal gradients against metrics of productivity like chlorophyll A, and then also a satellite-based estimate of iron stress. And this is the same iron stress proxy that Al and Nick were talking about. And so what we found was that there's kind of this mosaic of how certain regions where dissolved organic phosphorus accumulates as a function of chlorophyll, and some regions where it declines as a function of chlorophyll. What we found was that the DOP accumulates under conditions of alleviated phosphorus stress and iron stress. But then when DOP is consumed as an organic nutrient, it's really under conditions of enhanced phosphorus stress, but alleviated iron stress. So we found this unique link between the iron, the phosphorus, and ultimately the carbon cycle, whereby varying levels of iron stress in this vertical axis and phosphorus stress in this horizontal axis, leading to varying degrees of DOP being used as an organic nutrient or accumulating as a source of semi-labile DOP in one region of the ocean to in turn be utilized somewhere else. And we can, we've decided to further interrogate this organic nutrient hypothesis or test how these organic molecules are being used by phytoplankton. And so we decided to do some culture experiments, some culture work, and try and study the amount of phytoplankton phosphorus demand that is satisfied by DOP in some batch cultures. And so a graduate student of mine that just graduated, Caroline Anderson, this is her thesis work, where we grew Phytoceras and EHUX under increasing phosphate stress. So this is from 100 nanomolar to 700 nanomolar phosphate. And we supplied them 300 nanomolar of that glucose-6 phosphate, that labile phosphate ester that I showed you earlier on. And so what we found was that when you grow these phytoplankton under increasing phosphate stress, we saw more use of dissolved organic phosphorus that satisfy P demand at lower phosphate, and more elevated rates of alkaline phosphatases, which is what I'm showing you here in these graphs at the lower phosphate concentrations, which is not that surprising. But we were slightly surprised to see that even at this high phosphate concentration of 700 nanomolar, these two phytoplankton are using satisfying about 5 to 50% of their P demand from this DOP molecule. And this kind of surprised us until we dug a little bit deeper into the literature. We're reminded of this Orchard paper from 2010 in the Sargasso Sea from some field data where they flow sorted a bunch of cells that they were measuring the amount of phosphate ester uptake. And we see that the larger pico-eukaryotes and nano-eukaryotes satisfy a larger preponderance of their P demand from DOP than the cyanobacteria here. So maybe this is a way that larger eukaryotes can compete in these low nutrient ecosystems by targeting organic nutrients. And lastly, I'm going to talk about how we've been using some recent databases, compilations of dissolved organic nitrogen and phosphorus to build inverse models to simulate these data and diagnose the relevant biogeochemical fluxes. And so what the data tell us, this is for DOP and for DON on the right, but the data tell us in the dots and the inverse model output in the color mapping below is that there's elevated concentrations of DOP and DON in the tropics, and to a lesser extent the subpolar latitudes that decrease as we get towards the center of the gyres, which I'm calling here these organic nutrient streams, where the semi-labile DON and DOP is slowly consumed as they converge on 30 degrees latitude. Just some of the fluxes that we're able to diagnose from these inverse models, showing you the autotrophic DON uptake flux on the left, again approaching 40 millimoles of nitrogen, rivaling the nitrogen fixation flux. For DON, maybe a slightly larger preponderance or importance in the Pacific relative to elsewhere. For dissolved organic phosphorus, here I'm showing you the fraction of DOP-supported annual net community production of the biological carbon pump. Again, an important role for the Sargasso Sea, but also elsewhere on the Pacific basin. And we're able to do this inverse treatment, data assimilation for dissolved organic carbon as well, and so we can estimate the carbon to nitrogen to phosphorus stoichiometry of this laterally supplied DOM to the gyres. We find that it's N and P rich relative to the Redfield ratio, with some evidence too that the phosphorus is preferentially consumed relative to the nitrogen as this organic matter is transported to and within the gyres. Quickly, we also investigated this preferential remineralization question as a function of depth. And here I'm showing you the relative preferential removal of dissolved organic phosphorus relative to nitrogen based on the model decomposition rates that we diagnosed. And we see that there's this faster turnover of DOP relative to DON, and so this seems to be maybe a more important role for dissolved organic phosphorus relative to nitrogen. And so lastly, we're going to put this into our budgets that I introduced earlier. So we've got hot again on the top, fats on the bottom, add in these lateral nutrient terms, right? And we can now close or approach closing the budgets at both sites if we add in this lateral transport of here. I'm just showing you the nitrogen budgets. Case closed, right? Well, maybe not. You know, we can look at another tracer in this ecosystem. So we can look at when we look at oxygen and use that oxygen consumption below the euphotic zone, and then estimate how much nitrate should be accumulating based on the oxygen demand. That's what we call regenerated nitrate. Here I'm showing you a related tracer, preformed nitrate. That's the residual when you do this kind of decomposition. I don't want to get too much into the weeds of a preformed nitrate, but essentially there's this is from Ken Johnson's Nature paper from 2010, where this float that was deployed at HUT, where we have this large region of negative preformed nitrate just below the euphotic zone. And what this negative anomaly is telling us is that there's missing nitrate relative to how much oxygen demand has occurred. And so very quickly, some of our ideas of what could be contributing to this negative preformed nitrate is on the left. I got vertically migrating flora that are vertically migrating down to the top of the nitrocline to mine nitrate. And so it could be contributing to that missing nitrate. Or on the right here, maybe the nitrate is really not missing from the budget because a significant amount of net community production is channeled to carbon-rich, nitrogen-poor PEP or marine gels that somehow are contributing to the upper ocean biological carbon pump as well. So I don't have time to get into this too much. I'll just refer you to some of the literature. And I just mention it as a sense that maybe the total end demand and supply, those terms that I put in that budget are still a moving target as we kind of figure out the importance of these terms in the subtropics. And so to summarize, the subtropical gyres exhibit a similar biological carbon pump strength as high nutrient biomes, of which I've argued that lateral organic nutrient supply is one of the one of the larger terms in this budget. For dissolved organic phosphorus, it can supply up to 75% of the phosphorus demand in certain regions of the Sargasso, estimated from a model. And we found this unique link where it's iron alleviation from iron limitation coupled with phosphorus stress, which puts pressure on the DOP pool to satisfy autotrophic P demand. For nitrogen, it can supply up to 25% of the end demand. We know a little bit less about what specific molecules are involved and what are the kind of environmental conditions that are conducive to it being used as an autotrophic end source other than presumably end stress, so there's more work to be done there. And so what's next? I think we need more work on the organic geochemistry and what are the specific semilabile DON and DOP molecules that are involved, as well as coupling this to microbial ecology. Who is producing what of those molecules and who is consuming what? And I think there's an important role for biome state or the environmental conditions that favor organic nutrient accumulation versus preservation versus ultimately consumption as organic nutrient. I showed you maybe it's limited nutrient status, iron, phosphorus, but what else? Right on time, I'll just finish here with acknowledging my collaborators on this work and predecessors that have contributed to this topic over the years. Thank you.
Thank you, Rob. We'll have our next speaker, Harriet Alexander from WHOI.
All right, thank you so much. I'm really excited to be here today, and I think I'm one of the last talks standing between you and coffee, which is a good thing. Today I'm going to be talking a little bit about some work that my group's been doing, thinking about tracking phytoplankton nutrient stress across ocean basins. And I'm going to start out with an image which is near and dear to many of us in the audience. Marine photosynthesis. This is a plot of global chlorophyll across the world's oceans. And as we've heard from a variety of talks, it's integrally linked to the physical and chemical environment. And I'd be remiss after this morning's session not to say biological top-down controls, but I'm focusing more on bottom-up, so forgive me, virus people. And despite decades of study, we really have some fundamental gaps, as Al was showing in our first talk this session, in our ability to predict photosynthesis in the ocean. And so if we want to actually get to, oh, this is a different version of my talk, but that's fine. If we actually want to get down to how phytoplankton modulate biogeochemical cycles of carbon, nitrogen, and phosphorus, we need to get a better understanding of how they actually respond to their chemical environments. And ultimately, a goal might be to try to understand for various taxonomic groups, what are their patterns of nutrient limitation? Because I would argue that while we don't have a good understanding of primary productivity, our understanding of which organisms do well where, and also whether different organisms are experiencing the same sort of nutrient limitation is pretty limited. And a lot of my work surrounds the use of molecular tools. And there are a lot of different molecular windows that we can use to try to understand microbial nutrient responses. So here I'm showing a gradient. On the far side, we've got metagenomics, which is looking at DNA, down through metatranscriptomics, metaproteomics, and all the way to metabolomics, which is looking at the organic molecule composition of the environment. And each of these give you a different answer to a given question. For example, metagenomics is really great at answering questions of adaptation. We can look at gene gain or loss and get an understanding of these kind of longer-term adaptations of an organism to its chemical environment. By contrast, metatranscriptomics and metaproteomics give you a better idea about acclimation. So this gives you an idea of changes in the gene expression or shifts in the messenger RNA pool or the protein pool in response to a given environmental condition. Metatranscriptomics I like to think of as a very fine-scale, rapid response to a given environmental condition. RNA turns over very quickly, so this is kind of your fast response, what's going on right now, whereas the proteome is a little bit more of an integrated response. It has a longer turnover time, and so it can answer different questions. And today I'm going to be mostly focusing on the use of metatranscriptomics to try to understand nutrient limitation in the ocean. And a fundamental thing that we need to be able to do is actually identify genes that are nutrient responsive. A lot of this is based on homology to model organisms or organisms that we grow in the lab, and I'm going to highlight two things that have already been mentioned today. But one thing is the use of isolating organisms of interest and doing culture experiments and pairing that with some sort of omic assay. So here I'm showing some work from Sarah Bender, where she grew 3 different types of diatoms and looked at their response to nitrogen limitation. And one thing you can just kind of glance and see is the color indicates different expression patterns. These organisms are doing different things, which is interesting. So the pluses of this type of method is it gives you a very detailed metabolic view for a given taxa for what they're doing. And we can actually do a lot of work to couple this with really wonderful physiology, measure enzymatic rates, growth rates, et cetera, and get a good idea kind of more holistically what these organisms are doing in a bottle in the lab. However, plankton diversity is vast. So we can't do this for everything. There's no way that we're going to be able to do this for every organism that's living in the ocean. And also these experiments are very labor intensive. Another option is what Drew showed during her flash talk, which is a kind of field-based manipulation experiment. So building off of these kind of bottle incubation experiments, like Al showed earlier, you can collect a sample of your water, add whatever you're interested in. In this case, I'm showing an example where I added nitrogen and deep sea water, and then look at how that shifts either the transcriptional or the proteomic pool of proteins. This is great because it directly manipulates the organisms that are present in the field, but it has really high water requirements, and so it doesn't necessarily scale well across kind of larger cruises or other situations. So one kind of question that I have kicking around my brain is, can we start to leverage maybe larger basin scale nutrient gradients to identify nutrient responsive genes within omic sets directly? And today I'm going to be talking about work that I've been doing in a program called Biogeochemical. Biogeochemical is a program that's built on top of the GeoShip hydrography survey program. Here I'm showing a plot which shows all the different reference lines which are present within GeoShip. This has been running for several decades. And Biogeochemical has been, was initially started by Adam Martini's lab, and those initial tracks that his group ran are highlighted in blue, and we've since had a NOAA-funded pilot program which is funding the lines in orange. And Biogeochemical is a large team. It brings together some experts in omics, myself, Adam Martini, and Luke Thompson, as well as experts in optical and other measures such as flow cytometry, Jason Graff, Nicole Fulton, et cetera. And we are trying to integrate mature biological and chemical technologies and measures to inform different layers of the biological carbon pump in concert with these kind of fine detailed hydrographic surveys that GOSHIP's running. So doing a suite of omics from barcoding to metagenomics to metatranscriptomics, as well as bio-optical measures, imaging and flow cytometry, particulate measures, and acoustics. And together, this can give a really nice holistic image of what's going on in these communities. And these data have already been put to some really interesting use, and I thought that this figure from Adam Martini's group by Ustic et al. Is a really nice example of some of the questions that we've been circling around in today's session. They leveraged metagenomic data from across the world's oceans to identify global scale patterns of nutrient limitation in Prochlorococcus specifically. So it's colored by the different regions, and in particular, you can see some instances where they identify these kind of transitions or potential co-limitations within Prochlorococcus. And so I'm interested to see if we can actually expand upon this for other groups, because I would argue that it's likely that not everything is responding in exactly the same way. And in particular for eukaryotes, which tend to be slightly more complex in their genomic landscape and very important in an oceanographic sense. So today I'm going to be focusing on a transect that we did in 2022. It was the PO2 transect, which goes along 30 degrees north. It basically goes from Japan all the way to the coast of California. And we took a suite of measurements, optics, particulate C to N to P, HPLC, metagenomics, and my group was focused on the metatranscriptomic measures. And here I'm just showing the total chlorophyll across the transect, which is mainly to highlight that this was a super oligotrophic, very low biomass section of the ocean with a particular intensification kind of close to the coasts. And as I said, my group has been really focused on doing the metatranscriptomic analyses, and a fundamental problem with these kind of larger scale omic projects is scaling our computational tools. So a big focus of my lab group is the development of these kind of scalable and reproducible computational pipelines, and the work that I'm showing today relied heavily upon a tool that my grad student developed called Eurythmic, which takes raw metatranscriptomic data all the way through annotation and analysis. And additionally, the taxonomic annotation, which is with Ukulele. And for anybody who's interested, I encourage you to check them out. They're on our lab's GitHub website, and Ukulele is also installable as a Python package. So getting into the data. The taxonomic composition of our metatranscriptomes was fairly typical for what we see in oligotrophic data sets, in that it was dominated by dinoflagellates. This is not to say that dinoflagellates dominated the biomass, but rather that they have a very strongly shifted overrepresentation. Notably, we were only able to confidently taxonomically annotate around 30% of the annotated reads. So there's a large proportion of data which we can't annotate with any sort of high confidence. I think that this likely reflects biases in our cultured representatives and probably represents a lot of heterotrophic organisms. And this is an interesting space where we might be able to improve this with some sort of integration of metagenomic data. Additionally, we saw a really nice alignment with other measures. So here I'm highlighting a place where we saw a pretty strong increase in the proportion of the reads mapped, which were corresponding to diatoms in the eastern portion. And this aligns well with HPLC measures. So on the bottom, I'm showing data from the HPLC data, which was analyzed by Sasha Kramer at MBARI, and it's showing an intensification of the signal in HPLC for both diatom and dinoflagellate-related pigments. And for the rest of my talk, I'm going to be focusing on three groups, haptophytes, so encompassing Ameliana huxleyi and others, the diatoms, and the chlorophytes, or small green algae. Looking at PCA, kind of like what Drew showed earlier, we saw that they each had very distinct profiles in that they are doing different things, which is nominally what we would expect. But diving in a little bit deeper and thinking specifically about these kind of nutrient responsive genes, I decided to focus on pretty well characterized nutrient responsive genes. I pulled genes from a set of papers, which I've listed down on the bottom, and I'll go through them top to bottom. So for nitrogen, I pulled urea transporters corresponding to diatoms. For phosphorus, I pulled a glycerylphosphoryl diesterase, which is one of those enzymes which will cleave phosphate off of organic phosphate-containing molecules. Silica transporters, ISIT, which is an iron-responsive protein that's well characterized in diatoms. And then, because of the session that we're in and a little nod of that to Nick, I pulled the zinc transporter. And so you can look at this and see that broadly, we can kind of maybe infer some patterns of limitation. Broadly, what I would argue is that we're seeing a shift from kind of a primarily N-limited into an iron-limited system for diatoms. And in particular, the region farthest east, where we see this kind of slow increase in ISIP and a slow decrease in the urea transporter, I think is a pretty interesting region of transition for this kind of transition between nutrient-limited states. And we can go ahead and pull homologs in other organisms. So I've pulled in a haptophyte representative, and that's shown in the kind of dashed line, and then the dotted line is showing chlorophytes. And when we look at these data, the picture becomes a little bit cloudier, but roughly you can say that they aren't using these proteins in the same way. And this may be suggestive of differential nutrient controls on different groups. In particular, the iron, I think, is a kind of interesting example where we don't see any sort of induction of ISIP in haptophytes. However, the other potential answer, and I think is equally as likely, is that these organisms aren't necessarily using these proteins in the same way, nor are they potentially induced under the same sort of nutrient limitations. So it's kind of an open question. But if we take a more holistic look at this picture, here I'm showing a network analysis. So I took a gene correlation network, basically correlated all the genes and then identified major groups of those genes and correlated those with environmental parameters. And here I'm showing that for diatoms. You can see some of the major modules highlighted here, 1 through 5. And in particular, I want to call out module 1, which is genes that were associated with this kind of higher chlorophyll, coastally influenced environment off the coast of California, and module 2, which was a more dominant oligotrophic module that we had going across the set of stations. And module 1 was definitely enriched for genes which we know to be somewhat associated with iron limitation in diatoms, such as rhodopsin, FBA type 1, as well as ferritin, which is a little bit of a head scratcher for me. And module 2 had a kind of broader pattern of enrichment for a lot of genes associated with nitrogen limitation, as well as genes associated with phosphorus limitation, kind of getting at this kind of global growth restriction idea. So these kind of high coverage basin scale biological data sets that we're recovering from Biogeochemical as well as other programs, I think can begin to eliminate global patterns in nutrient limitation. And there's a lot of richness in this data. We can start to try to tease apart different groups, but there are inherent problems. We don't have a complete understanding of what different organisms are due. And in this example here, I was summing all diatoms together, and I know, or I have published a paper in which I showed that different diatoms in the same water mass are doing slightly different things. So there are limitations to this type of approach as well. In the PO2 transect in particular, I think it goes through a nice transition in the physiological ecology in diatoms, which we can document pretty well. And it allows us to get at an idea of the nutritional state of eukaryotic phytoplankton groups. And I think one thing that's really missing in our data set is the integration of trace metal measurements. I think that will really support the interpretation of these types of transcriptional studies. And I just want to highlight the Biogeoscapes program because I think that's an important next step. And then I think an even more important point potentially is that if we're going to continue to scale up these omic methods, we as a community really need to start thinking more seriously about intercomparison, as has been done within the chemical realm. So I think that this is a very important issue, and I just want to mention very briefly that Natalie Cohen, Adrienne Marchetti, Sarah Hu, and myself have been organizing a recently funded OCB working group, which is focused on an intercomparison of metatranscriptomic methods with an eye towards eukaryotes. Broadly trying to answer the question, how does sampling, collection, RNA extraction, sequencing, and bioinformatics influence our biological interpretation of these data? So we have a funded working group. We were able to get 12 participating labs, and Max Sato generously gathered a lot of filters for us on one of his cruises, which we distributed. People extracted the RNA, it's been sequenced, and we have 2 planned hackathons where we're going to be working on these data, as well as a series of bi-monthly webinars, which I encourage you to check out, which are focused broadly on kind of omic methods. And with that, I'm happy to take any questions. And I just want to highlight in particular Sarah Shapiro from my lab, who has handled all of the kind of high throughput RNA extractions from the Biogeochemical program, as well as Ariana Krinos, who's done a lot of wonderful work on the bioinformatic analyses. And thank you.
We are going to go into a set of lightning talks before our break, and Victoria will start us off once we're ready.
Hi, everyone. I am really excited to be talking to you about one of my favorite topics, which is how metal-containing enzymes drive marine microbial nutrient acquisition and biogeochemical cycles. And this is a project I had started with Julia Diaz at Scripps in collaboration with Rob Lampe from Andrew Allen's lab. And thanks to the great introduction from Rob, I don't have to convince many of you that dissolved organic phosphorus is really important for many oceanic phytoplankton, especially in times of phosphorus shortages. And ocean phytoplankton first need to produce hydrolytic enzymes, such as alkaline phosphatases that you can see here on the right, to gain access to this dissolved organic P pool. And those have metals in their active sites, such as iron and zinc. And now to gain insights into metals control DOP cycling in the North Pacific, I'm going to present shortly some results of incubation experiments that we conducted off of the coast of California on a cruise from an upwelling region offshore to the border of the North Pacific oligotrophic gyre. Here in the chlorophyll plot, you can see there was very high biomass in the upwelling region and a deep chlorophyll maximum offshore. Here we conducted shipboard incubations with the addition of iron, nitrogen, and combinations thereof. And these 2 plots represent the results in alkaline phosphatase activity as blue bars and chlorophyll biomass as orange circles. And we were really excited to see very contrasting trends where iron limited growth in the upwelling region. APA was low, but still stimulated by nitrogen. And offshore, we saw pretty much the opposite, where nitrogen was stimulating growth, but now iron was enhancing alkaline phosphatase activity. And to look in more detail into this phenomenon, with the help of Andy Allen's lab, we analyzed our incubation bottles for transcriptomics. I'm only going to focus on the offshore station with this higher use of DOP or higher APA here. So we were able to infer the community composition through metatranscriptomics, both in the, sorry, I lost my, here it is, both in the ambient seawater as well as in our incubation bottles. And we saw the dinoflagellates here in blue, haptophytes in pink, and pelagomonas here in green, where the dominant taxa present. And there's a really interesting shift in the bottles that were amended with iron. And now, since we saw in our growth experiments that both nitrogen and iron seem to somehow play a role, we looked for stress markers. And it is very interesting that we saw a very high abundance of nitrate transporters, indicative for nitrogen stress, which declined under addition of inorganic nitrogen. We also saw the presence of iron starvation-induced proteins, or ISIPs, and this iron stress was also alleviated with the addition of iron here. And those are just two examples. We have a panel of different nitrogen stress markers and iron stress markers that show similar things. But more excitingly, when we looked for enzymes that were able to degrade dissolved organic phosphorus, we saw that eukaryotic transcripts for alkaline phosphatases and for phosphodiesterases both increased in the incubation bottles that were treated with iron. And similar to what Drew said, this is a teaser and more work will be coming forward. But I find it very interesting that we can divvy up these transcripts by different taxonomic groups, and we will look more into who is using which enzyme and the response of different microbes. And so I just have a couple of take-home messages for you. We've seen that DOP is actively utilized and supports phytoplankton growth also in P-replete areas, that the phosphorus cycle is interwoven with nitrogen and trace metals, and lately that I would argue that we need more comprehensive studies, including but not limited to a combination of physiology, rates, and omics to really assess all these different stresses that we have on microbial communities. Thank you.
Hello, I'm Luai. I'm a postdoc here with Max Saito and Liz Kujawinski, but I'm going to present some of my PhD work with Aaron Bertrand at Dalhousie University and some fantastic collaborators at NIAS. Okay, so in this work, I'm going to present data from the Southern Ocean, where we did 2 incubation experiments. One was a bit offshore and one was closer to shore in the Weddell Sea. And in these experiments, we did a matrix design experiment where we incubated the microbial community under high and low iron and high and low temperature to examine how the microbial community reacted to these conditions. And we measured elemental composition, so macronutrients and trace metals, as well as metaproteomics. And the advantage of doing metaproteomics in this scenario was because proteins are the physical molecules that perform metabolism within a phytoplankton community. So if we measure the proteins, we get a window into metabolic rates. And also proteins often cofactor a lot of metals. So if you measure the proteins, we can also start asking questions about how the metals are behaving in a microbial community. Okay, so the first thing we observed was that warming and iron addition had synergistic effects on growth. You see here in this plot, particular organic carbon accumulation under different iron and temperature conditions. And what we see here is that when we increase the temperature only without adding iron, we only saw a very minor increase in growth. However, when we increased temperature and added iron, we saw a very large increase in growth that was more than the sum of the individual effects of iron and temperature. We've seen this before in cultures and in the Southern Ocean as well. And this could have implications for modeling primary productivity in the Southern Ocean, especially where there are many different things that are changing in the environment. And this suggests that there's some nonlinear effects on growth and nutrient drawdown that could begin to happen when temperature co-varies with nutrients. Okay, the other thing we looked at was trace metal stoichiometry. I'm going to only present on manganese, but if you come to my poster, I have a lot of other stoichiometries. And what we see here is that in the offshore incubations, increased temperature allowed the community to take up, or the community was taking up more manganese under higher temperature and added iron. In the nearshore incubations, there was a lot more manganese relative to nitrogen in the microbial community when iron was limiting relative to when there was a lot of iron in the environment. And that was super exciting to see because a lot of the culture studies that have been coming out show that phytoplankton increase their manganese and non-iron metal uptakes under low iron availability to maybe replace some of the iron-containing enzymes or to produce manganese-containing superoxides to combat iron stress. So moving from here, I wanted to ask some more questions about where was this manganese going in the phytoplankton community. This is where I use metaproteomics. And one of the proteins that contains a lot of manganese within phytoplankton is this manganese cluster within the photosynthetic apparatus. And what we noticed here is that under high iron, which is all the full circles that you see here, virtually all of the manganese that we measured was contained in this manganese cluster protein. So this meant that the phytoplankton community was investing a lot of resources into photosynthesis and growth. However, under low iron, even though we saw a large uptake of manganese, that manganese was not allocated to this manganese cluster protein. So this means that the manganese was going somewhere else in the phytoplankton community. And we have some guesses as to where that would be, maybe superoxide dismutase, maybe some of the phosphatases that Vicky just mentioned, and so on. Okay, yeah, so that's it. I guess, at this point, I just want to stress the advantages of using proteomics and metaproteomics to kind of help bridge the connection between omics and physiological rates, as well as elemental stoichiometry in the ocean. So come see my poster if you have any more questions or if you're interested more in this. Thanks.
Hi, I'm Marie. I recently got my PhD degree from Johns Hopkins University under the guidance of Dr. Nana Densenkan, and I will begin my postdoc journey at University of Washington at Seattle. Today, I would like to discuss a key aspect of my PhD research. The ratio of PIC to POC can play a pivotal role in the oceanic storage of carbon. We try to use random forests to compare the sensitivity of observed PIC and POC to environmental conditions so that we can better simulate them in the Earth system models. This work is in revision for GIL, and here is the latest preprint where you can find more technical details. This figure here shows you the variable importance for modest PIC and POC. We did this using random forest and permutation analysis. Large IMSE is indicative of predictors possessing greater importance, contributing significantly to the predictions. In our case, it's PIC or POC. We have 11 predictors here. Then we did sensitivity analysis to get a better sense of the underlying determinants of PIC and POC variability. We choose 4 predictors, and here is the partial dependence sensitivity analysis for each predictor. In each panel, you can see how PIC and POC respond when a variable changes from minimum to its maximum. For example, if you say, like we increase the mixer layer depth, we see both PIC and POC decrease, and then remain relatively stable. PIC is more sensitive. When ammonium concentration increase, both PIC and POC increase, and then reach a plateau, although we see a drop in PIC at the beginning. PIC and POC show different sensitivity to iron. POC increase, then reach a plateau, while PIC drops before increasing to even higher values. For shortwave radiation, we see they show similar patterns when the values are between 10 and 30. However, POC then remain relatively stable, but PIC increasing to even higher values. This second jump shows around 100. And then let me show you some special impacts of iron and light on PIC versus POC. We did this using the medium replacement analysis. The impact of iron on POC that shows really strong biomass separation at the equator as well as in the Southeast Pacific. Conversely, the impact of iron on PIC has less of the ocean showing strong effects. Interestingly, we see PIC sensitivity to iron is in the opposite direction compared to POC in the North Pacific, North Atlantic, and Arabian Sea. In terms of light, we see compared to PIC, POC shows less changes under the influence of light, and the biomass separation shows up at higher latitudes. This is in line with the fact that the sensitivity analysis of light. Okay, we have also begun our analysis on outputs from Earth system models. Here I show the variable importance for PIC and POC in MODIS, CESM2 and ESM4. We say for POC, shortwave radiation is the most important variable, although iron is not that important in both Earth system models. In contrast with the observations for PIC, again, all of them show shortwave radiation as the most important variable. Iron is not that important in determining PIC. This is in alliance with the fact that PIC is not as sensitive to iron as POC. Interestingly, we found that ESM4 thinks salinity should be the second most important variable, although we didn't see that in MODIS or CESM2. Although we think it doesn't necessarily mean COBOL model is incorrect. We are trying to figure out how realistic the calcite simulation is in ESM4 and give our explanations. Hopefully our work could help to improve the model to make better predictions of different kinds of phytoplankton on the climate change by including more precise constraints on biogeochemical cycles. Here's my to-do list. In addition to those, for my postdoc, I will develop variants of the cobalt biogeochemical model to investigate the impact of biotic calcification feedbacks on marine carbon dioxide removal strategies like OAE. I can relate what I have been doing to MCDR. I'm super excited about this. Thank you. Come find me if you have more questions.
Hi, I am Megan Sullivan. I'm a PhD candidate at UC Irvine, and I'm going to be talking about modeling variable phytoplankton stoichiometry globally. So as you all probably know, the elemental ratios in phytoplankton are variable, and at the global scale, this variability has some coherent spatial patterns. And we want to incorporate this variability in global models because it can have a major impact on the coupling of biogeochemical cycles, and what I'll particularly talk about today is carbon export. But it is a bit unclear how we can best parameterize this variability at the global scale. So what we've done here is embed a mechanistic cellular scale phytoplankton growth model for the C2P stoichiometry into this global biogeochemical model to constrain the parameters based on hydrographic observations. We're going to minimize the misfit between the hydrographic DIC phosphate alkalinity fields, and then look at what that tells us about the impacts on global carbon export. So first, we'll look at the right. And this plot is a zonal average of the C2P ratios of exported organic matter from the model. That dark line is the C2P from the cellular growth model. And what we see here, and then the red dots are observations of particulate organic matter from various cruises. And I'll note here that what we're plotting for the model is the export stoichiometry, which is not necessarily the same thing as what you would measure in the particulate matter, but it is interesting that they do have a somewhat similar pattern. And then on the right, we look at what the difference, how this impacts the model instead of using a constant C2P. So here is the change in the C2P ratio of exports if I had used the cellular growth model versus using just a constant optimizable parameter to have a globally uniform value. And so we would see if we had used a constant C2P ratio, we would be underestimating the export C2P in these red regions like subtropics and the Arctic, and overestimating export C2P in the Southern Ocean and upwelling regions in blue. And this matters because higher C2P implies more organic carbon export. So this plot that looks very similar to the one that was on the last slide is now that the change in total organic carbon export using our variable cellular growth model. And instead of using a constant. And from this, if we take a global integral, we would see that using constant C2P underestimates the globally integrated TOC export by about 3%, so a fairly small amount. But more interesting is the regional differences. And here, if we're looking at different regions, these changes in export are closer to the order of 100% changes. And this can be important for, especially when considering change, if we don't expect the changes to ocean fertility to be uniform spatially. And then the final thing that I will point out from this model is another interesting thing about this method is we can also look at the mechanisms driving C2P variability in more depth than if we had used just an empirical parameterization. So I'm just going to point out one thing that we found is that our optimization of the cellular growth model points to a major role of luxury phosphorus storage in controlling the C2P ratios. So here is the carbon quota in that cell and the phosphorus quota separated into the functional pool in the dashes and the storage pool in these dotted lines, and you'll see that the total is about the same, has about the same pattern as the storage. And if you have any thoughts about this or want to talk more, come talk to me by poster tomorrow morning.
Hi, everyone. So I'm Raquel Flynn, and I'm a postdoc at Adrian Marchetti's lab at the University of North Carolina. And I'm going to be speaking about the contribution of small eukaryotic phytoplankton to nitrate-based new production. So my study site is at Station Aloha in the North Pacific subtropical Gyre, and this study site is part of the Hawaiian Ocean Time Series. And what is interesting about this region is that it can be the station is that it's analogous for the whole of the oligotrophic North Pacific. And what we see in this region is that we get a two-layered system where we have the surface mix there where we have very low biomass, and new production is predominantly fueled by N2 fixation. And then situated at about 100 to 120 meters, we have this deep chlorophyll maximum where we see higher biomass and new production that is predominantly fueled by nitrate that is supplied from below, and it sits close to the nitrocline. So the goal of our project was to evaluate the contribution of small eukaryotes to nitrate-based new production and how these small eukaryotes respond physiologically to inputs of different nitrogen sources. So to do this, I conducted large bottle experiments where I collected water from both the mix there and from the deep chlorophyll maximum, and I added different nitrogen sources. So I had a control, one where I added ammonium, one where I added nitrate, and one where I added deep sea water. And I just want to make a note that the deep sea water addition not only had nitrate, but also had things like silicate, phosphate, and other micronutrients. And I incubated these bottles for 48 hours, and then I collected a suite of samples at the end. But because I don't have a lot of time today, I'm just going to speak about the phytoplankton group-specific nitrogen uptake rates. So what I did is after 48 hours of incubating, I then subsampled the cubitaners, and I did an experiment to look at nitrate and ammonium uptake. So I added an N15 tracer, and then I incubated them on deck for 4 hours and then did a filtration. And then I resuspended these cells and fixed them. I then took these resuspended cells and using flow cytometry, I sorted for specific phytoplankton populations. I then persulfate oxidized them and measured the delta N15 of these specific phytoplankton populations, which means I then have nitrogen uptake rates for specific phytoplankton. And I don't have much time, but I'm just going to speak about the proportion of nitrate-based production by the small eukaryotes. And I'm going to use the F ratio to do this. The F ratio is just nitrate uptake divided by nitrate plus ammonium uptake. And when we have an F ratio greater than 0.5, it means that net prime production was predominantly fueled by nitrate uptake. Well, when we have an F ratio less than 0.5, it means that net prime production was predominantly fueled by ammonium uptake. So if we look at the samples just from the DCM samples, and we look at the nanoeukaryote F ratios, we can see some interesting results. So when we look at the control, we see that they have fairly low F ratio, which means that in the normal environment, these phytoplankton are predominantly consuming ammonium over nitrate. When we look at the ammonium addition, we see an even further decrease in the F ratio. So they're consuming proportionally more ammonium relative to nitrate when they've been given ammonium. What's cool to see is when we add the nitrate, we do see this response in phytoplankton in consuming proportionally more nitrate relative to ammonium. And in the deep seawater addition, we see even more, a slighter increase in this response. And I didn't show this here because I didn't think I was going to run out of time, which I kind of am. But in the mixed there one, you can come look at my samples on my poster tomorrow, and you'll see that in the nitrate addition, we don't really see a response relative to the control. So we think that something else might be limiting them. So just as a quick summary, the nano-eukaryotes appear to be flexible in their nitrogen utilization, and they utilize nitrate when it's available and switch using ammonium when ammonium is available. When nitrate is available, the nano-eukaryotes dominated the biomass, and they seem to have a large contribution to total carbon export potential. And our goal is to take this data and couple it with metatranscriptomics. So look out for more. And if you want to scan to see my poster now, go ahead. Otherwise, I'll see you tomorrow morning. Thank you.
I would like to introduce our next speaker, who is Julia Diaz from Scripps. Take it away, Julia.
Great, thank you. Good afternoon, everybody. Thanks for coming back from the break. Hopefully you feel rested and ready for the next set of talks. Like Drew said, my name is Julia Diaz. I'm an associate professor at Scripps Oceanography, where I lead a research program in microbial physiology and looking at its impacts on larger scales. I'm also currently a AAAS fellow at the U.S. Department of Energy, and I've been there for about 9 months on sabbatical doing science policy work, and I will be going back after this meeting. So I wanted to take the opportunity today to share with you a combination of science and science policy as we work as a community to envision microbes as agents of global change. What are the federal resources and opportunities that are available to us through the lens of DOE? Okay, so I work in the Office of Science at DOE, which is the part of DOE that's the basic research part. So people are not always aware that DOE Office of Science is in fact the nation's largest funder of physical sciences research in the country. Folks are often surprised to hear that DOE is a leader in biosciences research as well, because you don't always think of bio and DOE going together. But as it turns out, ever since the Manhattan Project, which ultimately would give rise to DOE, there has been science focused on the role of radiation and its harmful effects on DNA. And DOE is continuing this legacy in DNA and genomic sciences with being a huge contributor to the Human Genome Project. There's a lot of biological research that takes place within the Office of Science under the Biological and Environmental Research Program. So many of you, I'm sure, have interacted with the Joint Genome Institute that serves as a major resource for sequencing capacity for the environmental sciences. The Biological and Environmental Research Division also has a longstanding legacy of funding research for bioenergy and biotechnology for the production of products and alternative fuels from genetically engineered microbes and plants. DOE also contributed to the development of all 3 COVID vaccines that were approved for use in the U.S. and also to Paxlovid, the antiviral. So on the other side of BER, the second half of this program is the Earth System Sciences Division, where there is an emphasis on studying environmental systems related to climate preparedness. And so this division primarily funds environmental sciences research within the atmospheric sciences, the aquatic and terrestrial and soil sciences, not so much ocean. But there is a little bit, which I'll talk about. But as a whole, these two pieces of the BER program function independently, and so there is a lot of policy interest to change that and to bridge these two parts of BER. And I think there is some opportunity for this community in thinking about how that might occur. So microbes have been proposed as sort of this bridge between the biological and environmental sides of research within the Office of Science. So in that spirit, we've heard a lot about microbes today and how they are important global engineers. I would include viruses in this, category, although they're not microbes, they're certainly biology and should be considered. And so I'm going to, for this first part, which is the science part of the talk, talk about my sort of perspective on this theme. So I think about ROS a lot. I think we all have our favorite topics in terms of, how microbes are important. For me, this is reactive oxygen species. And I'm going to talk about impacts of ROS production by microbes on the global ocean cycle. Part two is more of a call to action that draws on sort of the intersection between scientific innovation and public service, where, I believe that we have, a mission to contribute to the climate preparedness of society. This is a mission, DOE has been charged with, and, including microbes into that mission and incorporating that knowledge that we have in order to advance climate change understanding is going to help in that grand challenge. And so on that note, I'm going to be talking about some of the supercomputing resources that can be used as an opportunity towards that goal. All right, so I'm going to go into the science now. So we think of oxygen typically, phytoplankton produce half of the oxygen on the planet, right? And so we typically think of phytoplankton interacting with oxygen in that way. However, we also know that phytoplankton consume oxygen through respiration. Other microbes perform respiration to consume oxygen, but they also produce reactive oxygen species, and this can be a sink of oxygen as well. So reactive oxygen species are intermediates in the reduction of oxygen to water. There are 4 electrons in that transition, and with each one you get a different ROS. So we're primarily going to be talking about superoxide and hydrogen peroxide today. And all phytoplankton and bacteria that have been examined make extracellular ROS. This doesn't matter the degree of complexity that's represented on this slide, but just suffice to say there are a number of different sources of this within the cell, one of the primary ones being the production by cell surface enzymes that interact directly with oxygen that's present in seawater. So these are some of the rates. You can see a wide range spanning many orders of magnitude in extracellular ROS production by a broad taxonomic diversity of phytoplankton. These rates were all measured in the dark. And every time you make ROS, you are altering, the oxygen levels in your system. The question is, how permanent is that removal? So there was a study by a former MIT Joint Program student, Kevin Sutherland, in Colleen Hansel's lab, who approached this question. He scaled up the cell-based rates of extracellular superoxide production by these representative phytoplankton by their global abundance, and then summed the rates and came up with a gross superoxide production flux in the dark that represents roughly 36% of global oxygen production in the ocean. So this indicates kind of surprisingly how important superoxide is. 36% of all oxygen production passes through this intermediate. Now, when we're talking about whether this is a permanent loss of oxygen to the ecosystem, we have to consider the back reactions. And when you do so, we see the permanent loss is between 5 and 19% of gross oxygen production globally. Now, again, this was all based on measurements conducted in the dark. My former PhD student, Sydney Plummer, conducted a survey of 16 representative phytoplankton organisms in culture and examined across a light gradient how extracellular superoxide production changes. And what she observed was stimulation followed by saturation and photoinhibition for all organisms that she looked at. And we can model this with a modified version of a classic photosynthesis irradiance equation. So we think that Kevin's number for the oxygen sink is conservative. And we think this is under direct physiological control. This isn't just a passive process. So what we've seen is, under normal conditions where phytoplankton take energy from the sun to conduct photosynthesis, they produce NADPH and ATP. That NADPH is a source of reducing power that is routed to the carbon fixation pathway and other biosynthetic pathways. And as it turns out, the NADPH that's produced is usually in excess of what's needed in those assimilation pathways. And so a number of alternative electron sinks exist in phytoplankton to deal with that excess reducing power. And we found that the superoxide production flux is one of those mechanisms, because when we block the enzyme that makes superoxide using inhibitors in the lab, we see loss of viability, an impairment of photosynthetic health, and that is consistent with the buildup of NADPH and reductive stress in the cell. So we believe that this adaptive response could be triggered with further climate change impacts, with potential for changes to the global oxygen cycle as a result. And so it's been well established that changes in ocean circulation are going to occur with climate change. One of those changes is the fact that since the year 1970, there's been an average decrease in summertime mixed layer depths between 5 and 10 meters per decade. And so this could stimulate superoxide production by phytoplankton if this deepening is occurring within the photic zone and introducing phytoplankton into regions of the ocean that are sunlit and could stimulate higher rates of superoxide production than there were before. We don't know if this is true. It's a hypothesis, something that we are thinking about evaluating. Of course, the best way to do that is with modeling. So to bring it back out to the big picture, we all have sort of in our minds many different ways in which microbes can change the global environment. And I've listed a few more of them here. Another one that my lab thinks about a lot is nutrient bioavailability, and so phosphorus in particular. And it just so happens we're looking to hire and recruit someone to contribute to a study on phosphorus biogeochemistry. So if you or anyone you know is interested, please feel free to follow that QR code for more info. All right, so my dream as a microbial physiology scientist is to integrate the complexity that we know exists in microbial systems, right? That all the reactions that are happening in cells that we know have the potential to have global impacts when you scale them up, and to plug that into Earth system and ecosystem models to test, how impactful those metabolic pathways are. I know that there are some efforts being conducted towards this goal, but I think that there are many opportunities to push the innovation even further. So I would love to have conversations here at the meeting with anyone who's interested in this. I know from what I've heard, there are a number of challenges that have been identified to this goal. I think it's come up several times already in the meeting, just how complex and challenging such an endeavor would be in terms of collecting the data that's needed to parameterize these models, and coming up with the equations that are necessary to capture the full complexity of these processes. One element that's required, which I think represents more of an opportunity than a challenge, is the computing power that would be necessary to tackle this sort of goal. And so that brings me to the high-performance computing resources that are available through the Department of Energy. And starting out with a few definitions for those in the audience like me who aren't necessarily the most computationally minded. High-performance computing is essentially the same as supercomputing, and it is essentially it's not one big computer. It's a network of computers that work together by breaking up large tasks into smaller tasks that are computed in parallel. And we've been hearing so much about artificial intelligence, in the news and how it's going to change our daily lives. AI and machine learning is a type of computing that involves training models with supercomputers. So those typically go hand in hand, the AI and supercomputing. And exascale describes the level of computing power that we're capable of today. It describes a supercomputer that can perform 10 to the 18 floating point operations per second. That's an official unit called flops. So exaflops. And the DOE Office of Science broke ground in this field in May 2022 when Frontier, a supercomputer at Oak Ridge National Lab, reached exascale. And so this top 500 list is put out twice a year, where the supercomputers of the world are ranked. Frontier has held the top spot since 2022. And just this month, Aurora came online as the world's second supercomputer capable of exascale power. And so these computers are available for use by the research community on a no-cost basis. These operate at user facilities. You submit a proposal, and it gets evaluated by a review panel. And if your idea is accepted, your science gets to run on these incredible machines. By the way, it's the power of these machines is at least 50 times the computing power of the fastest supercomputer that's present on a university campus, which kind of gives you an idea of the relative scale. So this is a plot of market analysis that kind of gives us a peek into the future of supercomputing, into the future. So this is showing the major buyers of GPU, or graphical processing units. Those are used to power generative AI. So Microsoft and Meta dominate the market in terms of purchasing these GPUs, far over the level that DOE's largest systems are currently running on. And so the concern with this is, if industry is leading this trend, at some point in the future, these resources can become privatized, which would limit access. But with that challenge and concern in mind, late last year, President Biden issued the executive order on AI, which is the most expansive executive order that's been issued, reflecting the level of seriousness that the administration is looking at these AI issues. And so one of the provisions of this executive order was within 90 days of the executive order being released, that the U.S. government deploy the National Artificial Intelligence Research Resource Pilot, or NARE. And this represents a public-private partnership where 12 U.S. government agencies are working with leaders in industry to ensure that the nation's most powerful computing resources remain available to a broad community, including researchers. Not only research on AI, but research using AI. And so the question is, what can the research community do with these resources? And so I wanted to highlight E3SM, which is the DOE Office of Science flagship Earth System Climate Model. It is the only exascale Earth System model in the world, meaning it was designed to run on DOE's exascale computing infrastructure. And it's been a highly successful model, incorporating natural and human components of the climate system. It won a prestigious award last year at the International Gordon Bell Symposium for High Performance Computing for the implementation of a specific atmospheric cloud model that advanced our understanding of atmospheric dynamics in climate regulation. And just this year, it's expanded to include urban systems, which is really exciting because it can capture the benefits that are going to be coming to communities, especially underserved communities, when alternative clean energies are deployed. Now, one highlight, relevant especially for us, is the treatment of ocean biogeochemistry in this model, which highly underrepresents marine life. And so this is a schematic of essentially the biogeochemical ocean processes that are captured in the model. You know, carbon export. And I couldn't resist putting up this quote that was from the paper that sort of introduced this aspect of E3SM, which recognizes the limitations of this approach. And so highlighting, you know, the opportunity that we have to really enhance the microbial representation in this model towards our goal of climate preparedness. So instead of leaving you with a take-home message, I'd like to leave you with a take-home question, which is how will the ocean science community utilize the growing power of supercomputing resources? You know, I think even if you don't believe that AI revolution is necessarily going to change our lives, I think we all need to come to terms with the reality that the government, that industry is moving toward a state of prioritizing computational investments, and that could come with changes in funding opportunities and so on, that we would want to get ahead of or at least be aware of and maybe even shape as we move forward. So with that, I will thank my lab, DOE, funding sources, and thank you for your attention.
Yeah, we're going to hold questions for the panel, which will be coming up after our next talk. Our next talk is going to be by Patrick Rafter, who is at USF.
I have my puffy jacket on because I've come up here from Florida, and I am already acclimated only after 5 months. Thanks for putting together this great session on coupled biogeochemical cycles. Today I'm going to talk about coupling of iron and nitrate, but also coupling of timescales. So looking at paleoceanographic results and modern marine biogeochemical results. So let's see if I can use the mouse. Yeah. So our playing field today is going to be nitrate in the surface ocean. And so what I've plotted here is simply surface nitrate, and I've just colored everywhere where nitrate is more than 1 micromolar. Okay, so there are contours there that have numbers. That's the point here is to suggest or to show you that all these places where nitrate is persisting at the surface ocean are iron-limited waters. So you add iron and you increase nitrate utilization, primary production, and so on. So this leads to sort of the iron fertilization story, which is sort of top of mind right now, right? There's a lot of discussion about pathways for marine carbon dioxide removal. But the iron fertilization story is not just about iron, right?
Yes, interpretive dance.
Yeah, while we're waiting, I did want to conduct a survey. Have any of you downloaded a paper that one of your friends have published and then you never read it? You don't have to raise your hand, I know you have. I did that, and so Al and Nick, we should talk after my talk today. because I pulled it up, I watched your talks, and I was like, wow, oh, missed that. Yeah, it should lead to a nice conversation. Okay, so we're going back. Yeah. Okay, yeah. Okay, so yeah, the iron fertilization story is more than iron. However, I think, you know, casually you might think of it as sort of you add iron and a bunch of dominoes fall, and at the end of the day, we increase carbon flux to the deep sea. But yeah, it's more than iron, right? And some of the dominoes that need to fall are increased macronutrient utilization, like nitrate, increased carbon fixation, so on. Viruses are in there, bacteria is in there, all sorts of cool stuff. What it was a hell of a session today. I'm sort of reeling with ideas, really fun. It's late in the day. If you get tired or you have to leave, the more technical details of my talk can be found in these two papers. Just want to advertise that really quickly. Okay, so what we're going to do, so I told you we're going to couple timescales. One thing I'm going to do today is look at these ice age analogs for iron fertilization. And I should note here that we're going to use nitrate as the lens. We're going to look at iron fertilization, right? So it's sort of one of these first dominoes that should fall when you have iron fertilization. So we can look at, for example, what iron supply to the ocean looked like. And so here I have a plot from my friend Alfredo Martinez Garcia. It's from a site over here in the Southeast Atlantic. The x-axis is the last 4 million years, time going forward to the right, and the y-axis is the iron accumulation rate. And so what you see here is these peaks. Well, you see two things, right? You see this ramp up as the global climate gets cooler and cooler, and we have stronger ice ages, colder climate, drier climate, more dust, more iron fertilization, presumably. And then on shorter timescales, you see the ice ages, sort of these spikes in iron supply. And so you can see this pretty much globally. So this is a nice compilation. So there's citing Lambert and Cosentino's, which is a new compilation reconstructing dust supply globally. So you can see on the left sort of the current warm period, and on the right, the last glacial maximum. I'll just call it the ice age, the last ice age. You can roughly see, you know, it's larger than today. What? Oops. And so it's about, on average, it's about 3 times, the last ice age had about 3 times higher flux of iron, although different sites show different changes. And so we'll take a closer look at that in a second. First, let's take a look at just what did these global fields of nitrate look like during the last ice age when we had these nice analogs for iron fertilization. So equatorial Pacific, a place near and dear to my heart, there was absolutely no sign that iron fertilization led to more nitrate consumption or more primary production locally. So that's my own work, but also Cassandra Costa, Gisela Winkler, great results. I have unpublished work that looks over the last 5 million years. And if you look, here's the last 5 million years, you see a very big change over these timescales. We see no change in equatorial Pacific nitrate utilization over those timescales for the equatorial Pacific. So that's like a aha moment maybe. Looking at the sort of higher latitude Southern Ocean, we call it like the Antarctic zone. These are all records, by the way, almost all records of nitrate utilization based on nitrogen isotopes of marine microfossils and or bulk sediment. So looking at sort of the open Antarctic zone of the Southern Ocean, we see increases in nitrate utilization during the last ice age, but they predate changes in iron by something like tens of thousands of years. And the most likely explanation is that changes in upper ocean physics were driving those signals in nitrate. Looking to the subarctic Atlantic, you actually see less nitrate utilization. That's an interesting one by Marietta Straub. The Northwest Pacific, probably, yeah, it looks like there's some influence of iron fertilization on nitrate in this location, although
a little equivocal for the whole record, and also there's some pretty strong evidence that there are large changes in upper ocean physics, particularly that there was a kind of an expansion of the subtropical gyre that could also have a similar impact. And finally are the sites where we do see, I think, the strongest evidence for iron fertilization. Those are in the sub-Antarctic zones. So especially over here in the Southeast Atlantic, it's the strongest evidence we have. It's just one-to-one, you increase iron, nitrate, the nitrogen isotope suggests a big change in nitrate consumption. Just south of Tasmania, there are some coral records from our friend Tony Wong. Shout out to Tony Wong, I see you. Where it's sort of also like sometimes it looks like the fertilization is really happening, and then other times it's unclear. Maybe there's upper ocean physics playing a role. Okay, so we have set this. We have this playing field, the Ice Age playing field, and there's not a lot of green flags or green squares or even like green flags suggesting some of this iron fertilization was successful. So what's going on here? And I don't know where I'm going with this except to say that I just worked up this data last night, so that's where I am. I don't have a good transition here, but it's data from that Cosentino compilation once again. I'm just simply taking the ratio in this case of the Ice Age values versus the current warm period. All you need to know is like red is high, really high. And so you do see a pretty interesting relationship. The sub-Antarctic zones, sort of 40 to 50 degrees south, looks like they had more than a factor of 5 increase in dust supply. And funny enough, in the Northwest Pacific. So it does seem to be, and this is meant to be speculative, right? OCB is a place to put new ideas out into the community. I'm wondering about that. So okay, let's put that aside and just maybe the proxies for nitrate isotopes, for nitrate utilization, they're missing something. Maybe that's not the right way to do it. Maybe we should just have a general sense for global primary production. Well, in fact, if you're thinking about like the biological carbon pump, there is strong evidence that the efficiency of the biological carbon pump was stronger. So lower oxygen, I think this evidence is quite strong. Increased remineralized nutrients. If you need details for all this, I can go into it. Cadmium, calcium ratios. So together this is saying more remineralization of sinking organic matter. However, this doesn't necessarily mean that there was an increased flux of fixed carbon from the surface ocean, because there's also very strong evidence for a slowdown in the overturning of the global ocean. So this is some work I recently published. And also there's a strong case to be made that there was in the Southern Ocean, so the exchange of these deep waters that come up for a brief amount of time in the Southern Ocean, that was even briefer in the past. So sort of increased this air-sea disequilibrium. All right, but together, so we're talking about biology and physics. And altogether, we're at a point right now where, global fertility during the last ice age, not necessarily clear that carbon export from the surface ocean was higher than today, because there are these other routes to increase the biological carbon pump efficiency. So you see that sort of slight difference there. Okay, so we're at a lack of a consistent iron fertilization, and let's explore that a little bit. We're sort of getting hung up at the nitrate utilization step here. All right, so this is where I'm coupling my own work on paleoceanography with my own work in modern marine biogeochemistry. Also, once again, through the lens of nitrate and nitrate utilization. So this is a simple figure, cartoon of that I made up for the Eastern Equatorial Pacific iron-limited waters. And this is meant to convey that iron and nitrate come up to the surface, iron stays in the surface for a lot longer. And this is based on our, I think, very, very good constraints on changes in nitrate consumption over a 5-year time time span, actually. And I think pretty good constraints on what the iron supply to the system is. Overall, what we're finding is that there's nowhere near enough iron supplied to the system to explain the observed nitrate utilization on any of the like seasonal timescales I was looking at. So it must be preferentially cycled in the surface ocean. And so once again, technical details, you can look at these papers. I'm trying to give you sort of a young people's guide to iron and nitrate here, right figures and not too much complicated slides. All right, so to me, this preferential cycling of iron is not that surprising because marine ecosystems have had several billion years to evolve into this low iron ocean. And so I expect someone to say, how does it happen? And I will say, I don't know. The math is saying that it has to happen. We need to talk to the biologists like Randy Bundy. Okay, so we can skip this. It really looks like it's not just the Eastern Equatorial Pacific this is happening in. My back of the envelope calculations suggest it's in all the iron limited waters. Now, okay, so of course that cartoon is not accurate. This one's not fully accurate either, but it's a little more accurate. We have iron supply from the atmosphere, and we have loss from this surface ocean. This makes a couple predictions that are actually, the first is pretty hard to test, that there's this low sinking organic matter iron to carbon ratio. This is work that I hope we'll find these results at some point. Ben Twining's work is a really good way to approach this, I think. But another prediction that this system makes, and I'm looking for sort of independent predictions of this preferential cycling of iron, is that it suggests there should be a spectrum of iron limitation. And so I'm going to lead into the Browning paper, which I thought was just amazing, Al and Tom Browning, which I would like to meet. But independently, I predicted, okay, if this is happening, then when we're close to the upwelling zone, we should have sort of less, it'll be iron limited, but not so much. When you get off the equator, because you're not perfectly retaining that iron, you're losing iron along the way, it should be even more limited, okay? So this is where the Browning paper came in, and I can't remember which figure this was. It might even be in the supplements, but I was just like, wow, so cool. There's this range, this spectrum of iron limitation, and it's lower at the equator and higher off the equator. So this is about the ring, but I'm, special circumstances. So how do we couple these together? Maybe manganese. Maybe manganese is, what I should have been thinking about, and we'll talk about. But what I proposed is this sort of flywheel analogy for limiting nutrients. So it wouldn't just apply to iron. And so let's get back up and say, what's a flywheel? Let's say you have an engine, and the engine, all engines are pretty much going to have this intermittent power source. Let's say you have pistons. A flywheel sort of smooths out that power supply. So when you have too much power, it's sort of going to dampen that. And when you have too little power, it'll add a little bit of power. And it's just like a heavy wheel. So a heavy wheel, when I thought flywheel and I'm seeing this preferential cycling of iron, I'm thinking, well, maybe, this system has evolved to, in these low irons, microbes here have evolved for this low iron environment, and they're just a well-tuned machine for this. So you add a little bit of iron, and it's like, okay, cool, I'll slow down for my recycling of iron, but you won't necessarily see this increase in primary production, right? So that's what I'm proposing here. So I think that's kind of it. What we might be seeing here, how we might be able to explain the paleoceanographic proxy records, these analogs for iron fertilization, is that these sites where we see iron fertilization, they got enough iron that it kind of pushed beyond, it pushed beyond this flywheel analogy, and maybe even, change the community structure so that they could have much higher nitrate utilization, increased export to the deep sea. But overall, the analogs suggest we might not have seen like a globally much more fertile ocean during the last ice age. So my to-do list, I jotted this down right after one of the talks. Thanks and conclusions. It's pretty clear, I think, that the analogs are inconsistent with global sort of ubiquitous iron fertilization. And then we can potentially link this with this way of thinking about iron cycling in these iron-limited waters and to help explain that. So thank you, and I guess we're going to the panel. Thank you, everyone.
Okay, if we could have all of our speakers come sit up here, we'll move on to our panel discussion.
All right, you are not to be limited to these questions, but we thought, just in case the room was getting tired and you were out of questions, we've got some up here, but if there's anyone who has got questions for our panelists, comments.
Hi, Colette Kelly, I have a question for Nick. So I think of this R-star framework as a way of thinking about which organisms outcompete each other. And I was really curious with these kind of cascading, limiting nutrients, if there are likely to be changes in which organisms are outcompeting each other as well.
Yeah, thanks for the question. The trace metal literature early on, like 20 years ago, made this distinction between coastal phytoplankton and open ocean phytoplankton. And it's because if you tried to, grow the coastal phytoplankton in open ocean zinc or open ocean iron, they just couldn't hack it. So at some point, if you sort of extend that analogy, is that you couldn't grow Lactocyrus pseudonana at Station Aloha, even if you just gave them all the macronutrients and all the iron, they'd run out of something very soon. So I would imagine that leads to several successions. The models that we have can a couple dozen phytoplankton types if it's Steph's model, that, and maybe some of those we can sort of explore like what those successions might mean, and if there's like these ecotones with different sort of zinc susceptibilities or something like that that could be observable and tested even.
Test. Yeah. So this is a joint question for Harriet and Julia. I don't know about all the old modeling code out there. I suspect it's not optimized for GPUs, but in the bioinformatics side, a lot of the bioinformatics code, which is relatively new, is still not written for GPUs. And so I'm wondering what the community can do and funding agencies can do to help us develop the informatic tools that we need that are based around the new infrastructure so that we can take advantage of it. Seems like there's a bit of a mismatch there right now.
There we go. So I'm not aware of specific funding opportunities that would target that need. But, I do think that there should be a consideration of those factors at the policy level because, yes, it's not just something you could easily do to transition to the supercomputing hardware. So I guess I don't know of any policy work, but I can take that message back with me and have conversations. This is something that, I would love to talk more about, to get a sense of, this community's sort of needs and any talking points that you have that could be useful in conversations with the policymakers. I myself personally I cannot represent DOE in any official capacity, but, I know who to talk to and would be happy to, develop talking points, with this group.
I have so many thoughts on this question. I think that there's a major gap in our funding landscape for the support of scientific software and also the development of software, so fundamental algorithm development. I mean, I recently saw that SPADES, which for anybody in the room, is the foundational assembly program, is really no longer being supported. They're not going to advance it any further, and it's a labor of love by a handful of golden people who are keeping that afloat, and it's foundational to our ability to analyze metagenomes. So, yeah, I think we need more support for scientific computing software development, especially in the bioinformatics sphere. Maybe we should talk. But
Thank you. I'm over here. This question is for Patrick.
So,
So this was very thought-provoking. I'm old enough to remember John Martin's quote, "Give me a tanker of iron and I give you another ice age." So you showed that picture of glacial, interglacial glacial periods coinciding with high dust, right? So maybe you could just spend another few sentences on how you bring this into agreement with your data, on your findings.
Yeah, so the glacial periods were periods of high dust, in some cases, on average, about 3 times the current warm period. But some locations were quite a bit higher, almost 7 times higher, according to looking at the actual sediment records. And so what I think is happening, I think there's some threshold that these systems, because these systems are just so well tuned to living in a low iron world for billions of years. And so it takes a bit of extra power, extra iron to push these systems out of where they are right now. And where they are is what I argue is that they've maximized all of their, as a community, so it's got to include the viruses, the bacteria, the phytoplankton, that they've evolved to maximize the iron that they need, and so that they have it as long as they can. Yeah, so that's sort of, you have a follow-up question?
Yeah, so is there an alternative that maybe the ice age, the changes of the PCO2 does not have anything to do with an enhanced export? Because you mentioned also the reduction of
global climate, yeah,
and overturning circulation.
Yes. So if you slow down overturning circulation and you have, the same primary production of today, or maybe even slightly less, you're storing that carbon, and that's remineralized at depths for longer amounts of time, lowering atmospheric CO2. So I see your question was, connecting the dots to global climate. Yeah, and that's sort of where the community is right now, I would think. There's a great review paper by Danny Sigmund and Matas Hein that kind of go over this a little bit, although they did not cite my radiocarbon paper, and they should have, because it all works together, but it's still a good paper.
Hi, Yassir Edabar from Scripps Oceanography, and this is a question for Nick and anyone else on the panel who works on trace metals. Is there a consensus or general idea of how much is atmospheric deposition versus sources from the seafloor for some of these trace metals? And a second question is, you mentioned impacts of climate change on the supply of these nutrients. There's substantial interest now in mining the deep sea to support electrifications of transport. What should we expect from a removal of these trace metals from the seafloor on ecosystems?
Oh boy. Okay, here we go. I think I made a joke yesterday that based on what we were talking about manganese limitation, is that if you just did an iron fertilization in the Southern Ocean, eventually you would hit a wall and you would stop having productivity. But if nodules and ground them up and dropped them in the Southern Ocean, maybe buy enough of these other nutrients that production. So I guess I feel like we don't have a good consensus overall on the basin scale of how much is dust derived versus what comes up from below generally. I mean, Frankie Pavia in his lightning talk talked about how the models are really underestimating the dust flux in these low iron areas. And another thing that I think Douglas mentioned yesterday was the fact that, we're sort of already in the Anthropocene dust-wise or atmospheric source-wise. And so what we think of as like a change coming in the future, I think for iron, nitrogen, potentially other metals, potentially already here in terms of like our modern iron fluxes may not be sort of the pre-industrial state. And I think there's biogeochemical consequences that Douglas demonstrated really nicely yesterday. So I don't know how to do that except to be aware that what today is not like necessarily what the natural state of things are at equilibrium.
I can add something, maybe. From a modeling perspective, one way to answer that question is what sources do I need to include in the model to reproduce the data? And, obviously the cop-out answer is it depends on the element in question. Iron is clearly a combination of atmospheric, continental margin, seafloor volcanism. We know that we need models that include this kind of trifecta of sources, have better skill at reproducing observations. Other elements, maybe like zinc would be a good example. It's largely coming in through rivers. Cobalt, manganese, maybe, cobalt, I guess you could say, is margin dominated, I would say, very limited atmospheric deposition. But Nick raised an important point that, that we're also perturbing these sources. And so zinc is an interesting example. There's a lot of zinc in anthropogenic aerosol, right? So maybe that perspective would change over the Anthropocene. Cobalt is being used a lot, right, for various industrial activities, our batteries and things like that. It has a very small ocean inventory. So maybe it's very sensitive to mining and these kinds of things. So I think that's the challenge we've got, is that we're observing an ocean that's changing, right? It's changing in terms of its environmental properties, but also some of the boundary conditions are also changing. And some of the trace metals, that's a big challenge. But, it's also true for nitrogen. A lot of nitrogen coming into the ocean from anthropogenic activity. But this poses a challenge when we want to take a snapshot view of the ocean and say this is how it operates. It's actually a transient.
We have a question from the online environment. So this is from Elise Larkin. Question is, I would like to hear the panel's thoughts on the third question. What tools do we need in order to understand ocean basin scale ecosystem metabolisms, and specifically what observations are the most critical? Anybody?
I'll start. I'll talk for a bit and let everyone else come up with something more eloquent. But again, it depends, right? It depends on the metabolism that we're interested in. And I think it's a big operational, grand challenge. Rates, very difficult to measure. They're, very much of their space and time. So extrapolating rates is quite challenging, which has led us to try and use some other more integrative tracers that can help us along the way. In genomics is really has a lot of potential. But I always worry, a new tool comes on the block and, we can't neglect, it seems a shame to sort of neglect all the, large array of sort of physiological, classical, biological oceanography data sets that we have. I think what we haven't done very well is conduct kind of an experiment, where we actually try to deploy all the different tools in the same context. That's what I really love about Tom Browning's paper, is that he used the sort of oceanographic measurements of trace metal and iron and nitrate availability, bioassay experiments, and then the proteomics, and even up to the satellite. That required a smart guy like Tom, to think ahead that this would be a useful thing to do. Maybe an experiment would be really good. Make some traction on this.
I'll just kind of echo this point that Al brought up about measuring rates. One of the ways we traditionally validate Earth system models is we have tracer data, right? We've measured nitrate and phosphate and oxygen and a whole suite of other chemical tracers. And I think it was pretty well shown in Al's paper that Frankie showed us earlier in his lightning talk, that you can get the right answer for very different sources and sinks, right? You can match the tracer data. You can have a really high source and a really high sink to match it, or you can have much lower source and much lower sink to match it. And that's kind of, we have, net primary productivity rates are one of the things that are maybe a little more well measured that we use to validate our models. But I would say a suite of metabolisms. We need to measure the sources and sinks, the actual rate measurements in situ to help validate our models.
Hey, this is Tony Wang from Boston College. I want to come back to this transition from iron limitation to manganese limitation in the Southern Ocean, because it's come up in both Al, Dick, and Patrick's talk. So can you elaborate on that hypothesis, especially during the LGM scenario? So what's the iron to manganese ratio in dust, and what's the requirement in phytoplankton, and how would that change the productivity?
Yeah, so I think if you're just trying to do a stoichiometric comparison, iron is usually around 3% of like crustal materials. Manganese is like in the 500 ppm range or so. Usually a little bit more soluble than iron, but you're still not quite getting to that biomass requirement, which is pretty close to, the Southern Ocean diatoms are really good at driving down their iron requirement. So it's almost 1-to-1, 2-to-1, 2-to-3 manganese to iron requirement. So just imagine an ocean where all you're doing is adding dust to it. It's going to be manganese deficient relative to iron. I think one handle about the LGM that we don't have is interaction of coastal sources, because coastal sources are what drive the manganese cycle mostly, especially in the Southern Ocean. And we don't really know how well Antarctic margin sources were operating when they had a huge ice sheet on top of them. Maybe there's more erosion, there's more sources, and it's manganese source. But I don't think there's a lot of paleoceanographic reconstructions of the manganese cycle that we can pull from right now. But the data might be there.
All I'd add is that, yeah, I mean, I think that's for the LGM example. I think we haven't done any experiments with an altered ocean circulation. And, as Nick alluded to in his talk, the deep ocean is it's been more or less stripped out of all of its manganese. So, changes in vertical mixing, overturning, that could lead to some really interesting changes in the relative supply of iron to manganese that are then being sort of overprinted from aerosols, right? So actually we need to kind of get both sides of the story. I'd say that's a real clear and present knowledge gap for today, let alone for the glacial period.
Hi, Daniel Muir-Toy, CFA Institute. So in Nick's talk, we learned about in the absence of sufficient metal limitation, you can get driven to NMP limitation. And then in Rob's talk, we talked about the importance of organic sources. We know that the byproduct of phosphonate degradation will also produce methane, right? And so I have a question: to what extent do y'all think, as a research priority, with the increasing prevalence of metal fertilization as a geoengineering option, should we consider the question of, would we be trading CO2 for methane if we kick the ecosystem into NMP limitation?
Yes, I don't necessarily have a exact sense of what the methane production rates are as a function of DOP turnover, right? Because it's some of that phosphonate degradation that's producing methane and ethane. I have to dig a little more into the numbers, but when you also consider that methane is a much more potent greenhouse gas than CO2, it could, it's an important consideration that we've just kind of uncovered in the last decade or so, right? This finally uncovered this methane paradox. It's linked to phosphonate degradation, so it's an important consideration.
I can add one parenthetical to that, is that, there's also nitrous oxide. This was all, this famous LNO volume from 91 from the symposium. Methane was dealt with there, not from the phosphonate angle, but this idea that you crank up nitrogen cycling, or you generate oxygen deficits, and you produce other radiative gases. So I'm not sure the full sort of radiative counting is being done. My personal view, you don't sequester much CO2 from iron fertilization. And so maybe actually that means that you don't need to release much more methane or nitrous oxide to offset this small amount, given the big differences in radiative forcing between them.
Hi, this is maybe a question for Nick and Harriet. So Nick, I'll start with your talk in relation to, I think you alluded to the fact that there are microbes that don't require cobalt. I'm not sure if Prochlorococcus has Met-E, maybe it doesn't. So in that sense, it has an obligate requirement for cobalt. So if you had a scenario where we were doing iron enrichment, and so then you had more of a prevalence of cobalt, what does that mean in relation to the distributions of microbes and how we should be looking for that change as we sort of maybe shift what are the dominant limiting nutrients. And I'll have you maybe answer first, and then maybe Harriet, if you have a comment on that in relation to the omics, I would appreciate that.
Yeah, I'll just start this off by adding to what Colette had asked before about, how can we detect these transitions. And I think what Harriet was describing about, using these markers, if we can get taxonomic resolution, really high spatial resolution, we may be able to resolve those. Regarding Prochlorococcus as an obligate cobalt user and some eukaryotes that have evolved away this B12 requirement. There's an interesting paper from Yoshimi Ri in the South Pacific Gyre, and I think what she was showing using some single cell sorted productivity experiments was that the pico-eukaryotes, I think, are actually doing not so bad in the South Pacific versus the Prochlorococcus. And so they don't say this in the paper, and this is like a total overreach for me to say this, but you can imagine, I guess, that the pico-eukaryotes could start to take over some of the Prochlorococcus habitat. That might be a sort of an expected consequence of truly low and possibly low cobalt.
I don't think I have any additional fantastic thoughts on the issue, but I do think that the markers provide at least a window into that sphere, trying to dissect those cryptic.
All right, I'm going to take I have the mic, so I might as well ask my question. And it's not really fully formed, but I guess given the concerns that really when we started being able to do really good aerosol measurements of what trace metals are in aerosols was after we'd started greatly changing what the aerosols look like. Do you think we have a good sense of what I know we can do grinding up dust and seeing what happens when it lands in water, but that has its own challenges and issues. Do we think we're maybe not really sure what the composition of metals that was arriving with higher dust in the ice ages might have been?
Yeah, one thing I didn't elaborate on, but some really neat experiments that have been done. I've looked at putative glacial dust, so what would have been, and they found it was actually more soluble for, I think, all metals. So Elizabeth Schoenfeld did that work, really great work. That's one thing.
Yeah, I was going to mention she much more iron 2 in the dust. There's also been some work by Neil Wyatt and some guys in the UK who are looking at putative dust sources in Patagonia, and maybe found that there are some that are richer in manganese, to come back to your question, Tony. The big problem is there's a big difference between what you pick up, and Douglas, maybe I'll get this wrong, but there's a big difference between what you might collect at the source site and what ends up being deposited in the ocean, and that's the atmospheric processing, right? And so that's the big unknown, right? We might collect dust which has high manganese content. Maybe by the time it's been processed through the atmosphere, the iron has become more soluble, and that's a difficult nut to crack. I don't know if people are doing paleo atmospheric chemistry modeling. I suspect not, but that's a difficult one.
This is a question for Rob. So you talk about this lateral transport of organic nutrients from the continental margins to the oligotrophic jars. Have you thought about the transfer of the subsurface nutrients? For example, in the North Atlantic Ocean, there's this 18 degrees C subtropical water that got nutrient depleted from 200 to 500 meters during its transfer. And that would have significant impact on the nutrient budget. Have you thought about that?
Yeah, so in one of the figures I showed in kind of the middle of the talk where I was diagnosing DON uptake and DOP uptake that was laterally supplied into the interior of the gyres, I'm integrating over the annually mixed layers. So I'm integrating from about 120 meters to the surface. And I'm considering any nutrient that came from outside of that box, like 120 by whatever the gyre is. And so a significant portion of, I didn't show the inorganic nutrients. There is, a lateral supply of inorganic nutrients, and those are, of course, preferentially coming in those deeper layers because there's, not a lot of nutrients in surface waters. And so, I'm kind of integrating, I haven't really done and I have the output I could look into how much of the DON is coming in the surface Ekman layer versus the deeper layers. But again, it has a surface enrichment profile, so the opposite of the inorganic nutrients. So it's going to preferentially be in the surface waters.
Thank you.
Patrick, would more stratified and deoxygenated ocean contain, would it contain more iron in subsurface? I mean, from on geologic scale.
Well, no, I mean, yes, I presume it would be, but
right?
As we saw earlier from Frankie's talk, what the global inventory of modeling what the global inventory of iron is challenging.
Yeah, possibly. You might expect so. We see higher iron in oxygen minimum zones, but I think we're still, we're on that learning curve still with iron of understanding, is iron high in oxygen minimum zones because it's high margin supply, or is it actually that there's less, there's a redox signal that maybe you form less colloids because you can keep more of the iron as iron is. So you'd probably bet if you had to put 20 quid on it, you might say that there would be more iron, but whether that comes to the surface in your more stratified ocean is another matter.
Yeah, kind of a follow-up thought in a more stratified ocean under glacial conditions. What would be what would be the consequences for the nutrient supply then? Yeah, you have more stratified oceans, so you have less CO2 leaving the oceans through the mixing, but then you have less nutrient supply. So did anybody evaluate quantitatively what would be the relative effect of the nutrient supply versus lack of exhalation of CO2?
This is the theory that's what Pat just that's what we think is going to happen in the future as well. That the ocean is going to get better at storing biological carbon because it just has a longer residence time in the ocean interior, and this is totally decoupled from the export flux at 100 meters, right? So you upwell less nutrients, and so you can consume more of those nutrients, right? So you have less preformed, and then you
will be less upwelling also then.
That's right. So, but biological productivity is not necessarily directly correlated with the ocean's ability to store biological carbon, which is why the glacial, when we do experiments with our models with iron deposition, 10 ppm, maybe 15 ppm, the main hypothesis is that the lion's share is that the ocean just got better at storing soft tissue carbon, which is probably what's going to happen in the future as well, which is why we see that even though export production is predicted to go down, storage of biological carbon in the ocean will probably go up in the future.
Hi, I'm just curious about, I know that there's been a lot of work on mixotrophs in the Southern Ocean, but under this sort of prediction of future nutrient limitations in other ocean regimes, maybe this is a good question for Nick, and probably being inspired by Natalie, who's not here, so I'll ask for her. What do you think is the sort of future role of mixotrophs in a sort of changing ocean? Do you think they'll be forcing things in one way or another? Just sort of curious about that.
I don't know why this question is addressed to me, but thank you for it. Harriet, do you have anything? Oh man, mixotrophs. So there's a number of really compelling hypotheses about mixotrophs, how they might work differently in iron limited regions. What's it like to eat a bacteria for nitrogen versus eating a bacteria for its iron content? And honestly, I have no idea. But one of the things that I've realized is that as hard as it is to work with autotrophs in culture, working with mixotrophs is so complicated. Where there's people at UH do this, and you have to grow Proctococcus just to feed it to the mixotrophs and then keep them both alive. So I think they're going to be really challenging experiments if we want an answer to that.
think it needs like, I don't know, a real investment of people to try it out and see what it takes. Yeah.
We have one more question online, and then I think our buzzer is going to go off probably. So this is a question from Evan from HUI. His question is for Nick, Alessandro, and anyone else. So bouncing off answers to the previous questions about the difficulties of studying these nutrient limitations in the Anthropocene, does the community have a concept for how methods of NCDR that are bringing along these various trace metals might create varying responses due to the differences in regional nutrient limitations?
Okay. So, well, yeah, of course, right? If different so the question is asking about MCDR deployed.
Yeah, probably being deployed in kind of regional, environments, how, trace metals, either, I guess, with iron fertilization or even OAE maybe, might impact some of these regional co-limitations.
Well, clearly, we know that there's probably nowhere in the ocean where there's a massive supply of a secondary limiting nutrient, right? We think that's the case in the Southern Ocean, but that's just because we don't consider micronutrients that are limiting. So I think the big challenge is that in any system, once you add some of that nutrient, you're going to run out of something else very, very quickly. So how you go about identifying that, and then, yeah, you're charging around collecting manganese nodules and zinc batteries, chucking them in the ocean to kind of get you a bit further along. I think that's a big problem. I mean, and that's why for me, I'm quite concerned about this activity when we're talking about deploying these kind of human interventions when we actually really don't even understand what's going on. But we're learning, right? As we heard in all the talks today, we're still learning a lot about nutrient limitation, let alone some of the other more complicated feedbacks associated with our friends, the viruses, or the neglected friend, the zooplankton, who didn't get mentioned so much.
Yeah.
Yeah, I'll just add that I was involved in a study that used iron isotopes to estimate, how anthropogenic emissions are already modifying the iron cycle, and our estimates for that is around sort of a third to the iron that hits the North Pacific at least may come from an anthropogenic source. But in that case, we're sort of already, involved in some level of iron fertilization experiments, and yeah, I think it's hard to know what will emerge from a deliberate experiment over a long amount of time, but I think the likelihood that we will know the system well enough to say that there won't be any negative consequences, especially as you move up the food chain, I think is very low over the next 20 or 30 years. So hard to reconcile.
Thank you. Okay, I think, wow, we had some really good sessions today. There's a lot of similar themes too with capturing complexity and how do we scale that up and how it relates to all these biogeochemical cycles. So I know this was a long afternoon session, but thanks everyone for hanging in there. Let's thank all of our speakers one more time.